Right. Yes, yes. Unmute now.
రాలేదబ్బా.
Hello students, participants, kindly introduce yourselves. Akhilesh, mute your phone. Akhila. Hello students and also faculty participants. I request you all to mute your microphones throughout the session. You can inform your other classmates to join the webinar immediately. The program will start soon, as soon as our resource person joins. Okay. Students, you will also join the YouTube. Good morning, sir.
Good morning, madam.
Good morning, all. To the student delegates, faculty members, and resource persons of the day. Professor Gopal Krishnamurthy Garu from Bapatla College of Pharmacy. I welcome you all to this two-day webinar on research, methodology, and data analysis. Vijaya Institute of Pharmaceutical Sciences for Women, established in 2009 under the aegis of SRK Foundation, runs with the motto of knowledge is power. Recording in progress. So this motto drives the institution to take up various initiatives to disseminate knowledge to its faculty members and students. As part of this, today we are in front of you with one more webinar on research methodology and data analysis, the need of the hour, students and faculty members who aspire to become future researchers. The institution, in association with the Institution Innovation Council, is holding this two-day webinar. I once again welcome our resource person, Dr. Gopal Krishnamurthy Garu, and the other resource persons, Dr. Subramanian Garu and Dr. Prasanna Garu, who will be taking their sessions later today. With this, I hand over to our coordinator of the program, Dr. Praveen Garu, to address the audience. Thank you.
Good morning, everyone. A warm welcome to the two-day webinar on research methodology and data analysis. So in this context, today I will welcome the guest for webinar, Dr. T. Gopalakrishna Garu, who is Principal and Professor of Bapatla College of Pharmacy, located at Bapatla. Sir had obtained his graduation in B.Pharmacy from Lukman College of Pharmacy, Gulbarga, affiliated to Gulbarga University, and was ranked as the topper of the college since its inception in 1984. And Sir has obtained his master's degree in pharmaceutical specialization from Birla Institute of Technology, Ranchi. Sir has done his doctoral degree in the field of control release formulations from JNTU University, Hyderabad. Subsequently, he launched himself into the academic field as an assistant professor in the Bapatla College of Pharmacy in the year 1995 and promoted to associate professor and professor, from which he was elevated to the present level of principal in 2006. Sir has served as an academic supervisor to more than 60 master degree dissertations and 9 PhD degree dissertations for the award of MPharm and PhD.
మనది ఎక్కడ ఉంది ఇప్పుడు వీడియో ఆన్ చేయాలంటే
Sir is presently guiding 8 research scholars in different fields of pharmacy. Sir has published many research articles in various reputed national and international journals. Sir has shared his research experience on the platforms of 25 national and international conferences, workshops, seminars, and symposias. Sir is a member of professional bodies such as Indian Pharmaceutical Association and Indian Pharmacological Society Life Member. Sir is also an advisory board member of the pharmaceutical company Apogen Remedies Private Limited based at Hyderabad. Sir has also received various fellowships from Association of Pharmacy and Biotechnology, and Sir is a recipient of Meritorious Teacher Award from JNTU Kakinada. Sir is acting as editorial member for various journals, authored 4 textbooks, and filed for 5 patents. Sir acted as convener for 2 AACT-sponsored national seminars and 2 staff development programs. Sir also granted generous funds from AACTE under RPS and MODROP schemes during his academic service till now. So with this brief introduction, on behalf of Vijay Institute of Pharmaceutical Sciences for Women, we welcome you to the today's webinar, sir. Over to you, sir.
Thank you very much for your nice introduction and good words about me. At the outset, I wish to extend my sincere thanks to the convener, organizing committee, the principal of Vijay Institute of Pharmaceutical Sciences, and faculty members and my dear students representing various institutions. A warm welcome to today's session. So I wish to talk about what are different opportunities, challenges, or constraints involved in academic research. Because, am I audible?
Yes, sir.
Okay.
Yes, sir.
Okay. I will share my screen. Okay, my screen is visible.
Yes, sir.
Okay, thank you. So generally, the feelings of the students will be like this. As we are doing research in academic institution where the facilities are very less, whereas if you gone to some industry or some research lab, there will be good facilities and good research can be pursued in those institutions. In one way, I can say, in those institutions, if you are doing, there may be some opportunity for getting a good job in reputed pharma companies. But at the same time, you are not having some freedom, flexibility in case of the research organizations compared with the academic organizations. So you have to feel proud even you are doing research in academic institutions. And the research is not only restricted or limited to a pharmacy students or engineering graduates or a science students or medical students. Each and every human being, even in our daily life, we use it to do some research work. Even a farmer is also doing research work. Which kind of crop he has to cultivate, which kind of fertilizer, pesticide. And I'm sorry to say that even a beggar should also do a research work. Which place is ideal to beg? And a multinational CEO will also do a research work, how to expand the research, how to earn money In this way, each and every human being in their life, they have to do various kinds of research. So what is research? Research is a systematic inquiry to describe, explain, predict, and control the observed phenomena. So whenever we are doing a research, there will be some inputs and outputs. Output is our interest or our goal, for which purpose we are going to do that research. And that outcome may be influenced by some variables. And these variables can be controlled, and sometimes we are going to vary such kind of variables which may directly or indirectly influence your outcome. So how your results can be described by using which kind of table, which kind of graph. Of course, after completion of my lecture, you will be having the data interpretation by Professor C.V. Subramaniam sir. So by using this descriptive statistics, we can explain the things, and we can also predict. Say now everybody knows about this COVID-19 pandemic situation. And after next month, what will be the chances of cases? And in case of October, what will be the chances? So that is called as a predicted model. Diabetic prevalence. In 1990, what is the prevalence of diabetes? And in 2020, 2040, similarly, hypertension, various diseases and disorders, we can predict by fitting the data observed in various models. That may be empirical model, that may be mathematical model, that may be mechanistic model, that may be a simulation model. So we are having different types of models, different types of softwares, different types of programs like RLab, SPSS, or Minitab. So by using different kinds of softwares, we can predict. So that is definition of research. And there are different types of research. Namely, broadly, it can be classified into exploratory research, descriptive research, explanatory research. What is exploratory research? So, for example, pharma students use it to visit different hospitals where they are going to observe the clinical, social lifestyles of different patients. And say in that hospital, they are observing hypertension patients. And in order to treat hypertension, we are having availability of different drugs, different dosage forms. But what will be the adherence of the patient towards the schedule of the dosage regimen? For instance, the physician suggested the patient to take the tablet every 6 hours. Whether the patient is really taking that tablet every 6 hours or not. So that kind of research is called as exploratory research. It is unstructured. And in case of research, we can say the research is conducted in a structured manner and unstructured manner. Structured manner means it is a planned document. According to that planned document, we are going to proceed towards the research. And in case of unframed is unstructured. So that kind of explanatory research is normally unstructured. One of the example is just to observe the patient adherence towards the dosage regimen administration. And second one is a descriptive. Whenever you are doing even a simple experiment, there will be collection of the data. So that may be experimental data or a research data. And that based on that data, we are going to describe so and so research output is obtained from this experiment. So simple example, Beer-Lambert's law. You will describe the data in terms of concentration versus absorbance. So that is description. Whatever the data generated from your experiment, you are describing in the form of appropriate graph. That graph may be a pie chart, that may be a histogram, that may be a regression or linear graph, that may be a parabola. So which type of graph is better to represent your data is descriptive. And the other one is explanatory research. This explanatory research means, say, disintegrating agent is likely to influence the disintegration time or dissolution characteristics of a drug when it is presented in the tablet dosage form. So in order to explain that phenomena, you have to change continuously the input variables. So as the input is changed, outcome is going to be increased, whether it is having a positive effect Positive effect means whether the independent variable is increased, dependent variable is also increased, then it is called as a positive effect. Another may be negative effect. Yes, the independent variable is increased, the dependent variable is going to be reduced. So that is called as a negative effect. Whether you got a positive effect or negative effect, and how the dependent variable is fluctuated with respect to your independent variable, that can be found out by using explanatory research. So broadly, in this way, research can be classified into those three types. And my dear young friends, whenever you are doing a research, there should be certain requirements. And the research may be a qualitative one or quantitative one. And my personal opinion and my personal preference and my suggestion to all of you is go for qualitative research rather than a quantitative research. Qualitative research will be more appreciable and chances of getting good paper communication or publication will be more if you go for a qualitative research. And in terms of a qualitative research, we have to fulfill certain requirements. There are 5 requirements. One is valid. So what is valid means whatever the observation you got, the same you have to report, so that there should not be any manipulation, tampering of the data, because as a student psychology will be generally, if you have done some mistakes during the research by improper collecting of sample, improper analysis of the sample, then that time point they use it to fit the data by using some values. So such kind of data, such kind of, so that what I mean to say is you should be honest, you should be dedicated, you should have good commitment towards the research. Whatever you found, the same you have to represent. There should not be any partiality, there should not be any bias. So unbiased, founded, actually observed data try to present. Accurate. Accurate means whatever the observed values, it should be free from errors. That errors may be type 1 error or type 2 error, that may be instrumental error, that may be personal error. So there are different sources of errors which are likely to influence your experimental outcome. And you cannot design an experiment with 0% error or error-free experiment. But we can minimize the errors. Always try to do the research by try to control the errors as possible as you can do it. And reliable. Reliable means, say, doing your research, you may do some literature survey. And the same work, if you are doing, then if you got the same result, then you can say, yes, your seniors or the article which was published in that journal is reliable because you got the same data. So one of your senior student done that work. Your staff member may give that soft copy to you to do the continuation of that work. And if you are not getting the results which are reported by your senior, then what will be your feelings? Your senior is not done that work in a correct manner. That is why. So your reputation is reflected by reliability. Reliability of a researcher, reliability of any research organization will be reflected by your the reliable results. If you are reporting, then it will be stand sound. Next, timely. Whatever the data, because if you are doing a research work, there should be definitely a timeline. As if I finally So next, what are the opportunities in academic research? So one is a low competition, because in case of a industry, say a COVID-19 example, if we are taking, if every industry is ready to do vaccines or ready to develop different formulations to combat COVID-19, so the industry which is in front in that race, the industry which is going to come with a formulation or product to combat that formulation. So heavy competition will be there in case of industry. Whereas in case of academic institution, such kind of competition will be.
Sir, excuse me, sir.
Tell me, ma'am.
Your slides are not visible, sir.
Okay, okay. Madam, now visible?
Not yet.
One minute.
Okay, sir. Yes, sir. Okay, sir, now.
Sorry for the disturbance. Sorry for the inconvenience.
It's okay, sir. Fine, sir. You can go ahead, sir.
Okay, the various opportunities of research in academic institutions is the first one is a low competition. Yes, in case of industry, there will be a time bound, and the industry which is going to develop the formulation at earliest span will be having more appreciation, more profits. So such kind of targets will be there. Whereas in case of academic institutions, such kind of competitions will not be there. And collaboration. So in case of academic institution, some cases we may not have complete set of facilities. In that connection, we may visit some of the nearest universities or the organizations, government organizations like IACT, CDRI, CSIR laboratories, and they are going to support with a nominal cost, say in case of DSC, XRD, SEM, TEM, such kind of studies we can do by availing the facilities available in those organizations. Next one is a choice. So starting in your mind, you want to pursue some kind of research. Later, because of some constraints, you are not in a position to perform that kind of research, then you can shift to another point. So there will be some standby project you can keep. Whereas in case of industry, such kind of choices are not going to be there. They will give some target. You want to develop a particular formulation. So compulsory, you have to stick on to that formulation only. You are not allowed to shift to another formulation. And flexibility in terms of time. So you are not having any time constraints. Multidisciplinary. So in case of as in case of our colleges, there will be different departments. Say for example, cognitive, where they are going to isolate some active principle, or in a chemistry department, they will synthesize some drug, and later the discovered active principle or synthesized active principle can be collected by a physics person, where they are going to formulate into a suitable dosage form. Next, that can be transferred to analytical department, where the required analytical method can be developed for evaluation of that formulation. Later, the fruits of that formulation can be enjoyed with the pharmacological result, and so after analysis, it can be transferred to pharmacology department, where it can be pharmacological screening can be done, and reputation. So reputation of the academic persons will be shifted in terms of good quality of research work by publishing or presentation in various academic institutions in terms of conferences, proceedings, publications. Whereas in case of industry, that work is confidential, how much intelligent you are, but you are involved in development of several formulations actively, but your credibility, your reputation may not be allowed in the form of paper publications by the industry, as it is a confidential intellectual property. They are not allowed to reveal that work to outside that work. If you are doing that, then you will be behind the bars as per the existing IPR laws and regulations. So these are all different opportunities which we are having additional compared with the industry persons. So you can have good collaborations, and you will be having a plenty of choices. So these are all opportunities which we are having. And next one is constraints. What are different constraints which we are having in case of academic point of view? One is facilities. So facilities may be in terms of as I mentioned earlier, say in case of DSC, XRD, or in case of nanoparticles, Jetta size. So such kind of facilities may not be afforded by the self-financing institutions, though the faculty members will be expertised and having good intention to do such kind of research. But some facilities are lagging in our case of academic institutions, and other one is economical. In case of industry, they are not bothered about the investment, whereas in case of academic institutions, we are used to have a limited budget, and we have to complete our research work within that available economical budget which is allotted to our institutions. Of course, there is a possibility for applying funds to various the governing various apex bodies who are doing good funding to research like ACT, DST, DBT, CSIR. So that way you can try in order to overcome such kind of economical constraints. And other one is expertise and skill. So in case of industry, somewhat we can find more expertise, more skilled persons. Say if you want to operate some coating pan, some fluidized bed dryer, so that kind of operator, that kind of knowledge will be relatively strong in case of industry compared with the academic institutions. And other one is a team spirit. Of course, in academic institutions is also a team spirit. Of course, it is my personal opinion. Compared with the academic, in case of industry, there will be different teams will be allotted in order to develop, in order to design a new molecule, new chemical entity, or new product. So there will be a dedicated team allotted for a project work, and that team will be having healthy competition, because one team here is allotted to prepare, say, Reynolds extended release formulation. another formulation, another team may be allotted with another project. So these two teams will be having a good working environment, and such kind of team spirits, friendly relations will be more compared with the academic team spirits. Because one professor used to do one work and another professor is doing some cases overlapping of that research, and there may be some sort of unhealthy competitions. So that is the different constraints involved in case of the research pertaining to academic institutions. So these are all different opportunities and the constraints involved. So whenever you want to do research, there will be a need to go for this SWOT analysis or SWOT analysis. What are your strengths? What are your weaknesses? The strength and weakness are related to a particular person or particular institution, and hence they are called as internal factors. And the strengths and opportunities will be having always a positive impact. Because if you are having a good strength, not physical strength, what I mean to say is, of course, physical strength is also required to carry out the research, and the strength is in terms of research or in a particular selected research area, is a chosen, selected, specified research area. In that area, how much command you are having. Everybody will be having a fancy. I want to do some nanotechnology based research work, and I'm not having any previous exposure, previous knowledge, then automatically my strength on nanotechnology based research work will be less. So previous exposure will sound your strength. And weakness is again related to a particular person, particular institution, because research is termed to a person as well as institution. So what are the weakness? If you are having good committed student team, then automatically you can do a good research work, because research is always a teamwork. The strength is reflected by not only the supervisor should also have capability, and the students who are working in that supervisor should also capable of doing that kind of research. And though the faculty, so depending upon the mindset of the student, the faculty has to adopt the research and show
Sir, your voice is not audible, sir.
area we should be.
Sir, it's not at all clear, sir.
Okay. So next one is opportunities. Madam, my screen is visible?
No, sir, no.
Because some internet issue is there. Meanwhile, it is disturbing.
నీకు కాదు కాదు నీకు నేను మొత్తం 4 పెట్టే 4 ఒక స్కూల్లో నింది అంటే అది ఆ సీన్ నేను చేయింది పంపించా అది కాకుండా స్పెక్యులేషన్ అని దాంట్లో
Sir, the screen is visible, sir, now.
Whenever you want to perform a good research work, so you have to identify what are different strengths, weak points, opportunities, threats, or challenges. So strength and weakness, I already talked about it, and opportunities. So because of your research work, what type of opportunities you can have? Simply, a simple example is after completion of your graduation, if you want to go to MS, then if you are having a good paper, the chances of getting admission will be more compared with a student who is not having any research paper. And always opportunity is a positive and other one is a threat. Because of this research work, is there any threat? Yes. Say if you want to compare some marketed formulations, unfortunately, you may came to opinion that a particular brand is inferior. You are genuine, you've done that research work, and the results, whatever you want, you obtained. That results, the data showing that a particular brand is inferior. Then if you want to showcase a particular brand is inferior, then there may be a threat from that kind of companies. So that you have to take into consideration whenever you are doing the research work. So what are different strengths, weakness, opportunities, challenges? And next, in case of academic institutions, what kind of research work you can carry out? You can carry out 3 different types of research works. that may be historical research work, that may be observational, that may be experimental. So let me talk about historical research. For example, if you want to know about diabetes, what is the history of diabetes? Who is the first patient identified as diabetic? And what was the first medicament used to manage diabetes? What are the consequences of diabetes? And what was the treatment protocol in 19th century? What is the protocol followed in case of 20th century? And what will be the future possible expectations? So that is historical history. And what is the history of Asia Institute of Pharmaceutical Sciences when it was established, and how it is going to be improved from last 5 years, last 10 years. So in that way, you are collecting the data. You are not doing any experiment. Up to now, what are the information available in that selected area? So it is a type of literature collection. So based on the history of pharmacy, pharmacy provision, history of pharmacy education, history of pharma companies, pharma industries in our country. So that is related to historical research. And the next one is observational research. In case of observational research, you are not doing any attempt to alter your outcome. For example, a pharmacy student, as a part and parcel of their curriculum, they used to visit different hospitals. In that hospital, they used to collect the information, clinical data, prescription data from the bedside of the patients. So from that data, they may identify that particular age group of patients are having more diabetes, or particular weight group of people are having more diabetes. A particular lifestyle, particular habit leads to some cancer. So such kind of observation, you are not doing any research. If of course it is a type of research, but you are not doing any attempt to manage, to cure, to treat diabetes or hypertension. Just you are observing the data from the patients, from the treatment protocol, from the medicines which they are going to use. From that data, you are going to give some conclusion, some fact find that so and so group will be having more susceptibility towards a particular disease, particular disorder, so that you can do the research in your hospitals, observational research. And the third one is experimental research. So what is this experimental research is where you are deliberately, intentionally, you are altering your outcome by the appropriate changes in the input variables. So you want to replace metformin with glipizide, glycoside, or any other antibiotics, any other antihypertensive drugs. So by substitution of one drug with another drug, or by altering the dose, or by combining two drugs, is there any variations in the outcome, patient improvement in the lifestyle, in the health? So that is experiment. You are doing some experiment in order to improve the patient health by changing the dosage, by changing the drug, by altering the dosage, by correlating your lab reports with the drug dosage regimen. So that is called as experimental research. So in academic institutions, it is possible to perform all these three types of research work. You can do historical research work, you can do observational, you can do experimental studies. And some examples which you can do in your academic institutions. For example, the simple study is a hand sanitizer gel. So first you have to write what are the objectives of your hand sanitizer gel. So the objective will be like this: to design and develop an effective, safe, and skin compatible sanitizer gel. So that is our objective. And because of this objective, what you are going to learn from your project, that is learning outcomes. So what we will be having theoretical knowledge. In theory, you are studying water gels in technology one, in case of B pharmacy. Later, the theory is different, practical is different. Theory is easy, whereas in case of practical, you will identify what are different hurdles, constraints. and how to overcome the constraints. So practical knowledge will be always appreciated compared with the theoretical knowledge. So you can exchange your theoretical knowledge, you can transform your theoretical knowledge in terms of practical way. So that is transformation of the theoretical knowledge is possible because of such kind of experimentation, such kind of research work. So whenever you are doing a research work, definitely you have to communicate, you have to collect some chemicals from your lab assistant. So you have to talk with your research supervisor, your lab assistants and other colleagues. So in that way, you will be having increase in professional communication, leadership qualities. And by doing this practical, definitely our confidence is going to be improved. And you will be showcase your readiness to work in that industry. Because in the case of industry, they will ask what type of research you've done, what type of experiments you performed, what kind of equipments you handled. So that kind of data you can present effectively because of this kind of research work. That is advantages in case of students' point of view. Then you have to write what is the current status of gels. If the existing gels are superior or no pitfalls, no disadvantages, why we have to do another gel? As in the market already we are having some gels. So here you can find some materials like this. What is the present protocol used in COVID-19? The SMS policy, social distance, sanitization, wearing masks, and the sanitizations which are available in the market, they are available in the form of either solution form or in the gel form. And your objective is to prepare a gels. And normally the marketed gels are prepared with carbopol 934P, and the solvent is either alcohol or isopropyl alcohol. And the gels can also be formulated with other polymers, because in the theory we know that some materials like gelatin, methyl cellulose, sodium alginate, carboxymethyl cellulose, so these are all some materials which are going to form gels. Then why these are not available, why they are not used, why only carbopol is used? And if the carbopol is used, in order to form a gel, we have to use some triethanolamine or some alkaline adjustment of pH, and because of that pH there will be a partial neutralization of IPA, and the strength of IPA is going to be declined. So in order to prevent that, is there any other alternative substitutes for carbopol? And if you are using some hydrophilic polymers and you are dissolving the polymer in water, if you are adding alcohol, then there is a separation, that is a conservation of polymer from its solution by incorporating some non-solvent. And so what should be the steps in order to take, in order to overcome such kind of constraints, you have to write. Then you have to write your requirements. Whatever the gel you are going to prepare, it should be clear, it should be transparent, it should maintain effective concentration of alcohol, because in order to produce a sanitation, what should be the minimum concentration of alcohol and what is your target, and it should be organoleptically acceptable, because the consumer want more good smell rather than the percentage of alcohol. Percentage of alcohol is a legal aspect and organoleptical aspect is a consumer aspect point of view. And it should be stable and it should not produce any skin irritation, and it should be possibly approved by regulatory agencies. So these are all the requirements you have to write whenever you want to have a good proposal. And what is your outcome? What is your expectation of this study? Then you have to write what are the problems of existing sanitizer gels? Is there any strategies to overcome with small modifications in the existing composition, or whether you want to go for some other alternative polymers instead of carbopol? And what are different possible formulations in terms of composition, in terms of process? So that is the outcome of your research. In that way, the research work should be presented. And last, I will show one proposal. So how to write a design project? That may be by using the total change, TOC, or logistic framework analysis, LFE. And here in that proposal, first we have to write goal. What is our research goal? Even in case of academic, we have to write the goal. That goal should be clear because all the following work, all the subsequent steps are based on your goal only. If goal is not having any clarity, if it is having some ambiguity, you cannot perform a good research. Simply your goal should not be, I want to prepare some tablet. It is not the goal. You have to specify the tablet should contain how much amount of the drug, what should be the hardness, what should be the disintegration time. So that is critical quality attributes should also be mentioned. And what type of purpose or impact of a research. So here I showcased some example that objective is your development of some anti-diabetic herbal formulation. So what is the purpose or impact is your Indian traditional system is well known and having a vast medicinal plants, abundant number of plants reported to have medicinal plants. But unfortunately, all the plants are not standardized, scientifically studied. So there is a lot of scope. So to standardize Indian medicinal plant may be your purpose, or to develop alternative to synthetic anti-diabetic drugs, or to impact healthcare and economy. Because compared with the synthetic compound, your compound may be financially more affordable by the patient because your product may be cheap compared with the existing synthetic formulations. Next, with respect to each and every parameter, you should have some observable, verifiable indicator. Indicator means, okay, you are having some goal, some objective. Whether that object is fulfilled or not. As a teacher, we are teaching some concept, whether you learn that topic or not. In order to identify that, at appropriate intervals of time, a faculty member used to raise some questions. A similar way, if you are having some objective, at appropriate intervals of that research work, you have to identify whether the task is done or not. So what are different observable, verifiable indicators? So if you want to do some medicinal plant, then that should be authenticated by appropriate botanist, appropriate scientist. And whether it is having any biological activity, preliminary work was done, whether it is reported in our whole traditional medicine that it is having some activity. So that kind of check is required. Next, outcome. What is our expected outcome? Isolation of a new chemical entity is responsible for anti-diabetic activity because you are doing some extraction, and from that extraction you can go for some fractional extraction in order to isolate some active principle, not a mixture of phytochemical constituents. By isolating a single phytochemical identity, chemical entity, which is responsible for anti-diabetic activity, and other one is some processing the chemical substance into a suitable dosage form. So you isolated active principle. Next step, you can process it into whether it is suitable to formulate a tablet, whether it is formulated as a capsule or a liquid or a semi-solid, and what is the composition, what is your process, technology, which you can use to present your active principle into a patient-friendly, regulatory accepted pharmaceutical dosage form. That may be your outcome. Next, how you can observe this. So whenever it is a new chemical entity, whether it is a free compound or not, so it can be identified by using CNMR studies, by using IR studies. So what should be the spectral studies required to confirm what are different physical, chemical, microbiological, biological tests to be performed in order to confirm your component is having so and so structure, so and so biological activity. And other one is, next one is activities and resources. So what type of activities, what are different resources you are having? For example, how you are going to collect literature survey, and how you are going to do your solvent extraction, which method, whether you are going for simple infusion, decoction, maceration, percolation, even in case of a maceration, which solvent you are using. So that procedures you have to mention in case of activities and resources. And you have to write what are different assumptions involved, because simply you cannot start your research. That research should be having a backbone, that is assumptions. Why you selected that medicinal plant to have anti-diabetic activity? Because yesterday night you may not have a dream where the God is going to give some formula. Tomorrow you go to your college and you select a plant which is available in your medicinal garden, so you will be having antidiabetic activity. So that is not possible. So you should have example like flavonoids are having antioxidant property, and hence the any plant which is having flavonoids will be having definitely antioxidant. So this plant is reported to have antioxidant, and I want to test its antioxidant potential, which was not yet done. So that should be the assumptions. In that way, you have to write assumptions and what are the risks involved. Because every research will not be a positive, will not be a success. Definitely, there will be a pass and failure. And you may get positive results and you may get sometimes negative results. And negative results will also be welcome. Because say some cases you may have some assumption, that assumptions may not be always correct. And it is also a type of research. So and so plant, though it is having flavonoids, but it is not producing antioxidant activity. It is a surprising, because till now everybody knows that flavonoids are antioxidant activity. But a particular flavonoid is devoid of antioxidant activity. So that is a terrific achievement. So even it is a negative, because flavonoids are not proven now with antioxidant activity. So come forward, even you got a negative, say a binding agent is going to improve the hardness, but at a particular concentration of that binding agent, hardness is not increased. So come forward, you report the same, and that will be a really good quality contribution. Don't think that because earlier scientists have reported that as the binder concentration is increased, the hardness of the tablet is increased, I got in a reverse way. So if you got a reverse, you would repeat that experiment, go for number of replications. So second time, third time also you got the same, then you improve your confidence. So with that confidence levels, you can go with the debate with peer review, so that you will be having appreciated from the peer reviewers. So don't think about always you are going to get a positive result. And that is my suggestion to all of you, is whether don't bother about the outcome, but it should be having a well-designed experimentation with good protocol, appropriate documentation, whatever the results, unless you are not dedicated or committed. As long as you are dedicated, committed, sincere, and honest, then whatever the results, definitely you can fight with anybody with your quality of research. As you are having supporting documentary evidence, scientific evidence, statistical support. So all these are available, then you can go for even negative. Don't think about other way. So I think my time, because the second next speaker, Dr. Subramanian sir, is also ready, I think so. So I will restrict myself. So thank you, all of you, for your nice listening and giving me an opportunity to share some of my research experiences, which I am having and facing the constraints in academic institutions. So I wish to encourage all of you to go for either historical, observational, or experimental research. Whatever possible, you try to do. And after completion of your research, you try to communicate with at least UGC approved journal. Though in case of undergraduate level, it may not be possible to expect a Scopus or SCA indexed. So that is a constraint because of time. You may have good commitment, your faculty may have good exposure, but because of our time restrictions, sometimes we cannot do such kind of papers. Even then, you try to go for at least one nominal journal. You are not going to charge money, so that you will be proud. My article is published in at least some UGC journal. We are not demanding for money. Because nowadays, what is the problem is a BCS class 2 drug, which is not having permeability problem. But some of the research is used to study improvement of permeability of such kind of drugs. So such kind of articles are also going to be published by collecting some money. Then students will be in crossroad, because the faculty member don't encourage, faculty member is not going to encourage, because the drug is not ideal candidate for that research. But the student will feel that such kind of work was already published in a journal, but my faculty is not encouraging. So such kind of confusion status is there. So selection of ideal drug delivery system. So if you want to go for floating drug delivery system, first to identify which kind of drugs, which kind of diseases, disorders need floating drug delivery system. And you have to tune that your research to only such kind of protocol. So that is my sincere appeal to all of you, and thank you all of you. And if you are having any doubts, you can raise so that I will clarify. So it is a QA session.
Thank you, sir.
You're welcome.
Now,
madam.
I request the participants to interact with sir for any queries. If you have any queries, please come up. Either you can post them in the chat box also. Anybody there? So the session was quite informative and it totally fulfilled the objective of organizing this webinar on research methodology. You have very, in a detailed manner, clarified how to conduct research, how to have an objective, a goal, and how to do it with a kind of team spirit, how to go for negative results also, even they are open in research. For whatever suggestions you have given to conduct research in academic institutions, our students will come forward and take up those objectives. Thank you, sir. Thank you for enlightening us on research methodology with its opportunities and constraints. Thank you, sir. Thank you for being with us.
Welcome, madam.
Now let us move on to the next part of the session, that is session on interpretation of results in research by Dr. C.V.S. Subramaniam, Principal, Gokaraj Rangaraju College of Pharmacy, Hyderabad. I welcome our resource person of the second session, Dr. C.V.S. Subramaniam Garu, with a brief note on his academic profile. He has completed his M.Pharm and he has completed his research in pharmaceutics. He holds a doctoral degree in pharmaceutics from Birla Institute of Technology and Science. From Pilani. His industrial experience includes as graduate apprentice at Water Hindustan, Hyderabad, and he holds a rich teaching experience for 40 years. He is currently the head of the department pharmaceutics and PG coordinator at Gokaraj Rangaraju College of Pharmacy, Hyderabad. And he is also the principal of Gokaraj Rangaraju College of Pharmacy. And he also rendered his services as principal at Bapuji Pharmacy College, Day-1 from 2001 to 2004. And he worked as the vice principal from 1997 to 2001 at the same college, Bapuji Pharmacy College, Davangere. And He worked as the professor at College of Pharmaceutical Sciences, Manipal from 1991 to 1997. He worked as associate professor at College of Pharmaceutical Sciences, Manipal, and at College of Pharmaceutical Sciences, Manipal, he was also the reader from 1988 to 1989. And he has rendered his services for 6 years from 1982 to 88 at Birla Institute of Technology and Science, Pilani. And at Belgaum, KLES College of Pharmacy, he was the lecturer from 1981 to 82. He holds a rich research experience of 39 years, and under his research, 18 students have received their doctoral degrees and 45 students received their MPharm degrees, and BPharm projects were handled by 25 students under his research expertise. And Dr. Subramaniam Garu has published 40 international and national papers, and he has presented papers at 20 national conferences. And to his credit, he has the award of good teacher. He has been selected for this award by the students of College of Pharmaceutical Sciences, Manipal, through a secret ballot. And he has also received the Ranbaxy Research Award for Pharmaceutical Sciences in 1985 for guiding Professor R.C. Srivastava. He is a life member of Indian Pharmaceutical Association and also APTI life member and also life member of Indian Pharmacy Graduates Association. His professional activities include he is a referee for review of research articles for India Journal of Pharmaceutical Sciences. He is a member of editorial board Indian Journal of Pharmaceutical Education. He is also a member of Board of Studies for Pharmacy, Osmania University, member for Board of Studies for Pharmacy, JNU, Hyderabad. He has also rendered services as member Board of Studies for Pharmacy, College of Technology, Osmania University, Mangalore University, Mysore University, Bangalore University, Kuvempu University, Rajiv Gandhi University of Health Sciences, Bangalore. He has published various books and presented plenary lectures and also guest lectures at various conferences and seminars. Thank you, sir, for accepting our invitation and for being with us today. And to inform you, sir, one of your research scholars, Dr. Aparna, she is with us now at present.
Very good, very good. Right.
Thank you, sir. Now it is over to you.
Yeah, thank you very much. Good morning, every one of you. I welcome all the delegates to this webinar. I also pay thanks to the management of Vijaya Institute of Pharmaceutical Sciences for Women and Professor K. Padmalata Madam for inviting me to speak a few words on this particular webinar. Again, I will also thank the management, staff members and students for inviting me. Today, I am going to talk about what is called as interpretation of results. All these years, we have been doing interpretation of results in several ways during
Sir,
yes.
sir, excuse me, sir.
Oh.
Please, will you share your screen, sir?
Screen share.
Yeah.
Yeah, I have not yet started, but anyway, I will do that.
Okay, okay, sir. Fine, sir.
Okay. Is it visible?
Yes, sir.
Can I do full? Yeah, is it visible now?
Yes, sir. Yes, sir.
Fine. So all these years we have been doing a lot of research, etc. We have been doing interpretation, and interpretation is also being done in the practical class at BPharm level and PG level and research level. In other words, what I am going to speak to you today is almost everything is known to you, but I would like to highlight the way it is to be done. That means in a more systematic manner, such that we will not to leave anything, miss anything in the process, and take a very comprehensive look at all the results. This is the focus of today's webinar. So it is almost like everything you know and certain things we would like to add up. The objectives of this particular session is to appreciate the meaning and significance of interpretation. Basically, though we are doing it, but what is it exactly? The meaning of interpretation, how significant it is. Therefore, how we can pay more attention towards these aspects. Check, analyze, verify data for ascertaining the patterns. So underlying pattern or behavior has to be taken out from the data. Explain the importance of interpretation with reasons and examples. Several examples are there that we will know that how important it is, interpretation in a research or in regular practical class also. It is not necessary that everything will be covered only at the level of research, so that at B Farm level, at M Farm level, it will become a easy go. No. Describe the techniques of interpretation and explain the precautions to be taken at the end while interpreting the results. With these objectives, we'll move on. Meaning I have mentioned about significance, etc. Interpretation of results means what is the purpose? Why we should interpret? That means what is the end in mind? So keep that in mind. We have to go for interpretation. What is the purpose finally after doing interpretation? Did we able to get that particular expected outcomes or not? Importance of interpretation in terms of examples, both fancy as well as general knowledge examples, techniques of interpretation and precautions. Now, everyone has got a lot of data. There is no doubt about it. Say, for example, a college is going to NBA accreditation. Lot of data on students, lot of data on staff, lot of data on activities. What this data exactly means? When it is completely only data, how a common man can understand? So therefore, we have to come out from this particular data. Data is only the starting point, and finally, what is your contribution or conclusion finally? Maybe in one word or two words or two sentences or three sentences. That is more important than data. All data we have to interpret to get that particular outcome. So therefore, data will be there in NBA accreditation, even in teacher performance also, a lot of data is there. But what does this data mean? That is important. Say, for example, let us start with called a Tirupati Laddu. A Tirupati Laddu, a child, if you show, he may not be able to say whether it is Tirupati Laddu. Tirupati Laddu, first he will see with his own eyes and make a framework in the brain that this is something like a Tirupati Laddu. And this particular Laddu, there are several Laddus. Only the mother has to tell that Tirupati Laddu will be looks like this, eyes. So eyes is very first important aspect to recognize that particular one. So the same Tirupati Laddu, when the child takes nearer to the nose, he will get smell. Several laddus will have several smell, etc. But again, the mother tells him this particular characteristic smell is associated with Tirupati Laddu only. So he has got first shape, identification, he has confirmed with nose, with smell, etc. and then he put it on his tongue. Then he will taste that particular Tirupati laddu. So tasting is again a characteristic taste that is associated with the Tirupati laddu. When these three items are being felt by the child, then when mother tells that this is what is called as a Tirupati laddu, then he has to work out and associate what he has seen, what he has tasted with what is the words that a mother has given her. That is so. Touch is again skin. When you hold the laddu, whether it is Motichur laddu or Tirupati laddu or Bundi laddu, what are several types of laddus are there? In other words, all these tests, characteristics are to be confirmed one after another systematically. Then he will understand that next time whenever you show the Tirupati laddu, he will say, yes, this is Tirupati laddu. In other words, a child has been trained repeatedly on these particular aspects. And this is same as what is called as artificial intelligence. You have a software, you are training by feeding various data to that. And the computer learns, that is machine learning. So computer also was not knowing what is Tirupati laddu. Child also not knowing what is Tirupati laddu. So therefore, interpretation of all these things together is being done by previous experience, by data analysis. And definitely a pharmacist is a good interpreter because he does a lot of qualitative, quantitative testing, this, etc., pharmacopoeial testing. So all these things, after all these things, what he will say, this is pharmacopoeial standard of the drug. This is required. Well, after doing a lot of work for 2 years, 3 years, filing NBA, etc., finally, if you say that this is a standard, that is important for anybody to understand. Data has no meaning at all. Everybody has got a lot of data. But what is more important is the interpretation. Right? Okay. Let us see. We collect data, experimental, by doing experiments in the laboratory or by survey or several ways. If it is experimental data, 1 minute. You can have data collection from experiment. Yes, you have done laboratory experiment. Analysis by doing various ways of analyzing with simply instrumental methods of analysis or even theoretical. Theoretical is also possible data generation. Say, for example, in biopharmaceuticals, pharmacokinetics experiment, they have given only mono-exponential equation. You were asked to draw a plot. For doing a plot, you require time concentrations. But the question, it is there, it is not there. It is only given mono-exponential equation. So there you have to start with, when t is equal to 0, what is concentration? T is equal to 1, what is the concentration? 2 is equal to 12 hours, what is the concentration? So you have to generate data from theoretical. So this is how analytical or theoretical or experimental, whatever is there, the starting point is data. And data nobody can understand. Even experts like us also will not be able to understand the data unless you translate. Whether you interpret or you make me to interpret to understand. Just like the examination paper correction. Right? So interpretation is much more important than just doing. Skills are there, but doing. What is meant by interpretation? Analysis. Explore the commonalities, relationships, or processes. This is what is exactly called as interpretation. You have developed, compared your surface tension of ethanol with water. Is it high or low? Why is it low? Why is it high? This is what is interpreted. How much high? How much low? This is important interpretation. Anything, you take any standard plot or whatever is it, this Beer-Lambert's law standard plot, where it will not obey straight line. Where it will obey straight line. Is it really a straight line or a sigmoidal graph? So therefore, what is more important is this type of interpretation of the data has to be done. Interpretation of data. One aspect is verifying the hypothesis. So for example, hypothesis, you have taken a surface active agent. Now my question is your substance is really surface active or not? What is your answer? You will go to the laboratory, determine, put it in the compound in water, measure the surface tension and say, yes, there is a reduction in surface tension. Therefore, the substance is surface active. That is hypothesis. So verifying the hypothesis, for that purpose you are doing. Verifying hypothesis is a technical job. Interpretation. Prove or disprove whether this is really surface active agent or not a surface active. Work within this particular data. All drugs at one particular concentration, you will not be able to get further reduction in surface tension, that is above CMC. That means you have a working range. Below CMC, one type of behavior. Above CMC, one type of behavior. If you further increase the concentration, another type of behavior. Therefore, work within that particular data, we have to specify our interpretation by keeping an eye on that extreme as well as this extreme. And beyond data, what happens? So all these type of things, whatever you are doing interpretation, is highly technical. This is one aspect that you are doing it for the external examiner for M.Pharm dissertation. External examiner, B.Pharm project work evaluation. So this is one part of it. What is the other part? After doing interpretation of all the results, etc., you have to write a report. So report writing is very, very important contribution of results. If you don't interpret properly, you just can't write the report. You will just give a graph, you may give a table, but nothing. What is the significance? You have to draw inferences also. Again in the thesis. Drawing conclusions. Because you submit a thesis of PhD 500 pages or 600 pages. Now, for me, it takes months together to read the thesis. What I will do is go to the conclusions and see what are the conclusions. Normally, conclusions are 1 page or 2 pages. By looking at the conclusions, I can imagine what is his materials of methods. What is his literature survey? What is his introduction? In fact, all these things interpretation will come by looking at the title itself, by looking at the thesis itself. You just don't have to read the entire things. You can take a white paper, keep writing what he must have done. If he has not done, that means there are gaps in the thesis. So you have your own judgment on these type of aspects. So therefore, drawing conclusions is very, very important. And when you want to tell others that you have done a work and this is the conclusion, that is one technical. Many times we also come across non-technical people. For them, a broader meaning is required. Don't tell all the data, graphs, etc. I don't know. Finally, you tell me what is your conclusion? What is your meaning of your thesis? You have to explain 3 years of work in 3 sentences. Can you do that? Generalizations. A new knowledge, as it has contributed to any new knowledge, as your M.Pharm thesis has contributed any new knowledge, or has it confirmed the existing knowledge? Yes, it is true. Or deviating from the existing knowledge, why is it so? So it should have all thesis, all research work, all interpretation should lead to a newer knowledge. If he is not adding newer knowledge, then you can't call it as a research. Somewhere some fault is there. Fault with interpretation, fault with the experimental, or fault with the design itself, or fault with the objective, etc. So therefore, a theoretical concept may develop, may not develop, but it has to be done. So what I would like to say is, whatever is written on the red, it comes under this particular part, that is conclusions. What is the meaning of your work to the public? You will not do, you should have a communication. What is communication? You write a report, you write a M.Pharm dissertation, and you submit. That is communication. Communication to external examiner, give me A grade, give me B grade, etc. So you are writing all these things in order to get your grade or marks, that is evaluated. And if you publish the data, you are helping the scientific community across the world with your intellectual capabilities. That is interpretation. Now, what is the purpose of interpretation? That means ultimately, what does it mean? This is important. That means we are trying to have end in mind doing all this interpretation work. Identify usefulness or utility of that particular results, data. The abstract principles that work beneath the findings must be studied. What is the abstract principles? For me, mathematics is an abstract principle. But I should be able to understand. Statistics is another abstract principle. Several theories are there. Newton's first law, Newton's second law, or what is all these thermodynamics, first law, second law, then Dixon-Crowell cube root law, Beer-Lambert's law, all these are laws, abstract principles. Fresh inquiries can test these predictions later on for the future work. For example, I will take second law of thermodynamics. When second law of thermodynamics is proposed to the scientific committee, people never believed that, because it is there only on a theoretical platform. It is the imagination of the scientist. Until you test this particular theory in the laboratory, it will not be accepted. So, the acceptability of abstract principles we have to study. The first law of thermodynamics theory has become a practical, and later on it has become application starting with steam engine. In steam engine, what is the efficiency of a steam engine? All of you will study in physical pharmacy. Nowadays, the syllabus is not there, but all your teachers knows, all research scholars knows. So therefore, when steam engine efficiency is being calculated, then they have understood second law of thermodynamics. Until then, it is abstract only. But we have to work with that. Second example is refrigeration. You call any refrigeration engineer to your college and ask that I want to refrigerate this air conditioning of this particular hall, seminar hall. Immediately, he will measure height to distance, volume of that particular one, how many people will sit, how much of people's perspiration, how much AC should be there, whether it should be 2 ton or 3 ton or 4 ton. All these are being done by thermodynamics only. Heat of enthalpy. This is again the starting, but today, even now, anybody, if he goes for refrigeration and air conditioning, he will always talks about the FPS system only. Because thermodynamics are started with thermodynamics FPS system in particularly European countries. Right? So therefore, there are abstract principles today. Tomorrow, there may be some applications for that. It may be very, very important. So let us say mathematics and orthogonal functions are very useful to us. Today, we use orthogonal functions in design of experiments and optimization. But what is that we have done is using orthogonal functions. Therefore, what is abstract today has to be studied to understand the behavior and its application. And this is end in mind we will be studying. Similarly, optimization, formulation of statistics. If you say that Taguchi, what you say, no, Taguchi and all these people, he is a statistician worked in the pharmaceutical industry. His job is to reduce the defects of tablets or ampoules or whatever is it. And his statistics he should use there. Today, what we study, the total quality, TQM, quality management, or ICH guidelines, Six Sigma approach, benchmarking, business reprocessing activities, all those hi-fi activities are started from the statistician only. All quality standards, quality assurance we are doing now, studying, all those are started by statistics only. And these statistics have given quality improvement, process control, etc. Today, you go to a market and purchase a refrigerator, fridge for your home. He will give 7 years guarantee. Can any? He will give 8 years guarantee. Can anybody give? You purchase a washing machine. Normally, warranty means 1 year. Guarantee means 2 years, 3 years. Say, for example, you purchase a car. That fellow will give lifelong guarantee. Because they have faith in that quality. So therefore, statistics have got an important role, and it is definitely abstract principles. All these abstract principles work for that, and ultimate aim is what is the purpose of these things. So abstract principles that works beneath the findings must be studied. Second one is explanatory concepts serve as a guide for future works. What is the explanation? How a concept is being developed. It opens in new areas, several new areas. I will give one example. Aspirin, where it will be absorbed. Every B Pharm student will say it will be absorbed from the gastric region, because aspirin is an acidic drug. So when first it is being studied in the research, they have studied aspirin only and got an interpretation that acid, because it is acid, it is absorbed from the gastric region. Okay, if this is correct, then my ibuprofen also should be absorbed from the gastric region, because that is also acid only. So after doing 2 experiments, you have come to a state where all acids will be absorbed from the gastric region only. Tomorrow, I will give one example of acid. Immediately you will say it is absorbed from the gastric region. How can you say? Have you done that absorption test? Have you gone to the laboratory? Have you done? You have not done. But how can you say? Because having done 1, 2, 3 times, you have understood the underlying philosophy, explanation. Is it acid? It will be absorbed in the gastric region. Is it base? It will be absorbed in the intestinal region. This is all what we have studied in biopharmaceutics and pharmacokinetics. So therefore, there is no need. Whatever research you do, it will help the people like this. Tomorrow I will ask, where from penicillin will be absorbed? Because almost everybody knows penicillin is an acid. That is also from gastric region. So a person who has not done that today or tomorrow, I will give him a base. For example, chloroquine, where it will be absorbed without doing experiments, he will say it is from intestine. Because explanation is offered. What is happening in this particular one? Continuity is being maintained. Having done the research work and underlying principles are known, explanatory concepts are developed such that tomorrow you need not to do the experiment, directly you can offer explanation. And this is the power of the education. This is the power of the knowledge. And this type of interpretation we should know. Continuity of research can be maintained by this particular one. You take any acid, you test, it will be automatically true. It opens new intellectual adventures. This is very important. So it enlightens the intellectual to imagine what will be the future course of action. As far as we are concerned, biopharmaceutics, pharmacokinetics, we have studied from textbooks. Several scientists have prepared and kept things ready. We have to try, read, understand, apply. That's all. Interpret. Graphs, whatever is it. You go to the pharma, students go to the clinical pharmacy, hospital, and you apply. Interpret. Because you have got the principles. This is the type of interpretation required, correct interpretation. I will give you few examples which stimulate quest for knowledge, strive for knowledge. For example, when I studied my B Pharm, I have studied only H1 antagonists. I have never studied H2 antagonists. H2 antagonist was not known in 1977. Later on, receptors have been identified. What is the first drug? Cimetidine. Cimetidine work has given us the H2 receptors with proper interpretation, results. That took long time. But if you take ranitidine next series, it will take only 1 year, 1.5 years only. And it has become so strong. And today you don't see cimetidine in the market because properly being understood the research. Now famotidine. So like that, one tidine comes, all tidine series follows. So the first one is the most important one, which gives explanatory concepts, serve as a guide for the future work. So therefore, this example will not stop there. There are several examples. For example, propranolol, all lalas. You have so many lalas in the market, all hypertension. First one is very, very important. That has given the explanation to the world. All other things take very small times. You take fluoroquinolones. First one is naradexic acid, or otherwise called as narofloxacin. Today, you have a flood of floxacins. So many number of floxacins had come because explanatory concepts are here. And so new fluoroquinolones came into the market, which has killed the earlier the thought-breaking products. This is through explanation. You don't stop there. There is another example. When I studied B Farm, I have never studied proton pump inhibitor. Proton pump inhibitor was recent origin. The first one is omeprazole. Then you have a lansoprazole, pantoprazole, so many prazoles had come into the market. Because explanatory concepts are well developed. Receptors are developed. Targets are developed. They are properly understood and explained, interpreted, tested. All these things are done. That is why there is so many number of drugs had come into the market. Third one is appreciate the findings, why and what they are. This is important. The findings means data, graphs or tables, whatever is it. You have to appreciate what are the underlying phenomena. What are the causative factors? Others understand the real significance. In this particular context, I will make one statement. Data is more important. A data, if you cannot understand, you cannot interpret, no problem, publish data. Some other person will interpret. It is not necessary that you have to do the data, you have to interpret, you have to understand, you have to explain. Sometimes, if you can explain, very good. Otherwise, data has got a credibility. So even now, in several instances, data has got credibility. M-Farm thesis, we say that you submit the data. Let your supervisor understand it. Let the external examiner understand it. Put it on the paper. Let the world understand what is this data. But the data should be correctly being done. So data has got a value. And further interpretation has got an interest. So therefore, others understand the real significance of research. You don't judge for yourself. Don't go for interpretation, because the whole scientific community is there behind you. If you have got credible data, there are people to help you for credible interpretation. And based on this, the theory can be developed. Are you with me, all of you? Yes? Participants?
Sir?
Are you here? Are you with me?
Yes, sir. Yes, sir.
I will give one. Yes, I will give one more example. Say, for example, in Diwali, we will do a lot of crackers. These will go into the sky and give you different colors. What is happening? How colors are being obtained? Okay, when you were first B Farm class, you must have done flame test. You keep the substance near the flame and through glass you will see what is the color. Based on that particular flame test, you will say it is sodium, it is potassium, it is calcium, like that you will write qualitative analysis. Now here you are able to understand only the qualitative. Why can't you convert this qualitative into quantitative? You know it is sodium is there, but how much sodium is there, how much potassium is there? So therefore flame photometer has come into existence. Just a qualitative turning into quantitative. Appreciate the findings. Today, after flame photometer, now we have got AAS, atomic absorption spectrophotometer. And this particular one, all metals in the periodic table it will analyze. If some gold is there in mine, so they will bring and do it in AAS. Gold or silver, whatever is it. So a simple observation in the laboratory will allow us to expand why, what they are, and how we can take them to further heights. A flame test, atomic absorption spectroscopy is being developed. Color tests for SS. There are several qualitative tests, say all colloidal tests. or inherited test. You do qualitative test or take it into quantitative measurement of absorbance, etc. Principle remains the same. So the findings are there. It is how we will expand them further. And this can be done by right interpretation. Say, for example, I will give one more example. All of you know these examples. It is not that, but we are trying to recall them. Say, for example, Van Hoff factor. In physical pharmacy, Van Hoff factor comes under qualitative properties. Depression in the freezing point, elevation in the boiling point. We will make the students to do the experiments in the laboratory. So the entire work has started by Van Hoff only for non-electrolytes. Somebody has got doubt. Is all are non-electrolytes? There may be electrolytes also. This is the origin of thought. Therefore, non-electrolytes is the starting point. Then you have electrolytes. Then electrolytes have become strong electrolytes, weak electrolytes, dissociation constants, everything has come up. This is what is called appreciated. Why, when, and how things will definitely matter. Okay, so with one particular thought, by proper interpretation, a series of steps have come. This is what is called as appreciate the findings, why and what they are. Exploratory research. And exploratory research is one particular area which also require what is called as interpretation. This particular example illustrates you how good the data should be. We take the example of Boyle's law. Everybody will study in physical pharmacy. When Boyle has done this particular law, he has done only PV. He never knows what is T. But he has done gases. Boyle's law, lot of data. Take X gas, take Y gas, data. Gas. But what are the commonalities? Then when commonalities are there, he has proposed this. Similarly, Charles also took the other 2 points. He also studied all the gases and he established the relationship. This data is available in the research community for longer time, maybe 40 years, 50 years. It is the third person, one more person that he has to take this particular data and bring it into, combine them into PV is equal to KT or PV is equal to RT. Today, what you say ideal gas equation PV is equal to nRT is based on that particular data, exploratory research that the other people have done. This person joined all the loose links and brought it into an equation called as ideal gas equation. Now, when ideal gas equation has come into existence, people have got a doubt. Why can't be ideal solutions? Why can't be ideal solids in solutions? Yes. The thought is ideal only. So the philosophy, what is being proposed by one, started continuing. Therefore, you have got a Raoult's law, ideal solution. You have got a Hildebrand's Scatcherd equation for solids in solutions. So all these things, ideal gases, ideal equation, liquids in liquids, ideal equation for what is called solids in liquids, ideal gas in liquids, that is also there. All of you study this in physical pharmacy. All of you know these things. The interpretation is the transition from exploratory to experimental research. So first they have got only ideal gases. They have started experimenting solids, solutions. They have started experimenting solids. Then they got. That is what is called as exploratory research. These provide later on theoretical concepts. All these theoretical concepts of Raoult's law you will use in steam distillation, in pharmaceutical engineering. It has got a lot of application. But what is more important is the exploration of all these, one concept leading to several, one drug leading to several drugs, etc. Purpose of interpretation. Performance index. In a staff performance index, annual performance index of staff members. All of them will give 18 classes. This work, that work. What is more important is whether you have to give, you have to measure them first of all. That is what is called as metrics. US FDA has come to such a level, but even every activity has to measure the metrics. A good example what NBA accreditation has given. Metrics is a good example where all your activities have been converted into numeric values by NIRF. So when you want to go for promotion of sales, incentives, again interpretation, training, whether training is successful, pre-test or post-test, or off-test, on-site test. How is it successful? Again, interpretation of data. Product comparison, weather forecast. Weather forecast is a very important interpretation. This particular rains have not come for last 100 years. Today only, recently only it has come into the news. That means this fellow has got the data for 100 years and he understood, interpreted each cyclone, each weather forecast from different parameters and angles. And because of that, today he is able to forecast floods 48 hours beforehand. Forecast cyclones 48 hours beforehand. That is interpretation. Now you can understand that how strong is the interpretation that is allowed. Product comparison, BA/BE studies, identifying equivalency. Reference product, standard product are equal. What is that? Interpretation. Improving performance. There is a product in the market which is better. My product is not that much better. How I can improve my performance? Again, interpretation. Take the parameter, take up levels, etc. Optimization of parameters. Optimization is a full chapter, unit that is given in 4th year, 2nd semester, research methodology and statistics. Again, interpretation, testing. Drug-drug interactions, avoid adverse interactions. It is an interpretation. UV visible spectrophotometer, FTIR, NMR, mass. What do they do? Identify a particular chemical. That's all. Authenticity is verified. If it is not authentic, you can't use them in the production. So therefore, interpretation of data is very important. Correct or not. Biological experiments, where a lot of variability is there. The animal experiments, human experiments, all these things, fixing the dose, fixing the dosage regimen from animal studies. And that data you have to extrapolate to human beings. In mice, this is the dose. What is the dose in human beings? Again, if this is interpretation of the data and extending to the other things. So again, it is important. An excellent example. X-ray diffraction. Without X-ray diffraction, particularly physical forms, whether it is polymorph or whatever is it, or same crystalline structure, US FDA prescribes X-ray diffraction is the only technique to identify it is the same polymorph. Therefore, batch to batch variation will not be there. Preparing ranks, NIRF ranks, etc. analysis of laboratory results is an excellent example of interpretation. Now, you go to a clinical testing laboratory, you give your blood. Next day or same day, he will give a report. You read the report. What it will tell? He will say, this is this much, this is this much, this much. Can you really understand what is that? So he will say above, below, average, normal. Okay. Then try to correlate that, interpret your data with that, and finally say that this is normal, this is above, and this is below. And more important thing in any clinical reports is at the end he will write correlate with the clinical symptoms. If it is to be correlated with the clinical symptoms, why to pay so much money and get the report? That means whatever report he is giving is only a numbers. It is the doctor who has to interpret. You have got some idea because it is normal, above, and all these things. So this is a classical example where data is there, but its interpretation is a technical job. And it is important. And this is exactly the reason why we will go for one opinion, second opinion, third opinion, and finally, what is called as public poll in Facebook or Google or WhatsApp. Right? So blood pressure versus age, the relationship. Differential diagnosis is a very good example of interpretation. Cough, normal cough and cold cough, TB cough, any type of, several types of coughs are there. All these coughs are not same. You have to differentiate them. Interpretation is required. Biological products, sameness, that is, biological products means one is biologics and another is biosimilars. These are again comparisons. And you see, they say biosimilar. US FDA will have a different type of language for interpretation also. It will never accept that it is a biologic, biosimilar only. Better is also there. How? What? Meaning. But they say it is better. You have to interpret. Production of failure, batch failures, etc. is again an interpretation. So what I mean to say, all these examples will tell that there is a lot of interpretation is required in any decision making. And decision making will be only yes or no. Whether professor is given or not given, only yes or no. We has got a lot of data, paper publications, all these things. You have to come to one particular mark. What is the annual performance index, API? So establish, explore the concepts within the data. This is important. When you have conducted 5 experiments or 6 experiments, is there any link between these or not? This is important. Concepts throughout the data. Okay, you have changed binder. What is its impact on the tablet hardness? Next, friability. Next, disintegration. Next, dissolution. These are all some parameters. So you have to have the link between all these parameters, all the tests. If there is something wrong in between in this particular link, the entire concept changes. Because you know, any of the philosophy, any of these phenomena will be not always linear. It goes up, comes down. You have to identify the peak. It decreases and again increases. Up to a particular angle of repose, it will increase. After that, it will decrease. After a particular size, flow properties increase. Afterwards, decrease. So everywhere, this is the phenomenon. It was never same as that one. Either it is biphagic, or otherwise it is sigmoidal, or it is log linear extension. Any of these. We should be able to understand that. Linkage of one result with others is very, very important. Each experiment has to be linked properly with interpretation. And that is important. Otherwise, if there is somewhere in this particular thread, if something is broken, then entire you have to repeat the experiment. Establish continuity. This is continuity from this thesis to next thesis, continuity of this work to the next work. Everywhere there should be certain amount of continuity. Because I heard some PhD students will say that I have completed the work, there is nothing else that can be done further. But frankly speaking, if nothing else can be done in that PhD, it is not worth PhD awarding. Because each work has to link. That is why in PhD thesis, they will say last heading is future work. If you cannot imagine what should be the future work, then nothing can be end by itself. Nothing can be end. You write your conclusions. You may win the external examiner, but that is not the correct one. I have done everything, sir. It is almost like a student telling, I have studied well, but in the exams, what will happen? We do. So whenever you do formulate theory into hypothesis, this is important. What is hypothesis? Hypothesis is a relationship, that's all. If you increase the concentration, OD will increase. If you increase the concentration of surfactants, reaction will increase. If you decrease, this is what is called as hypothesis. And whenever you plot the data, it will be linear or it can be log linear. Sometimes you will get curve. I can't interpret because it is curve. If it is a straight line, you would have been happy because I know it, because it is a straight line, extrapolation, interpolation, very precise. You have to make curve into a straight line. Until you get a straight line, you take log linear. If you don't get straight line, you take log linear. Emerson's log linear is being done. Therefore, any type of these type of equations, linear, log linear, you have to construct them and understand curvilinear and biphagic relationship. Most of the natural processes are biphagic. As you increase the partition coefficient, absorption will increase. After some time, it will decrease. Any parameter you take, it is like, and almost all parameters are curve only. You take time curve, you have to take log, then it will become straight line. Then you can do the parameters. If you just cannot get a straight line, you cannot get any PK parameters. So you should have this particular techniques of interpreting and making them straight line, or looking at the equation, draw a graph, looking at the equation, tell me whether it is a straight line or not, looking at the equation, positive slope or negative slope. All this is interpretation only. Dissolution studies, angle of repose, particle size, one linked to the other. How these things are related. It has to be done in a proper. Now, techniques of it. This is what I have tried to tell. Now I am going to talk about techniques of interpretation. It is not an easy job. That is why many people will fail. Probably that is why we want to discuss more in detail. It is a skill. It is a dexterity. It is desirable. Several times it happens. A student will go and take IR. What is drug excipient interactions? He has to do, interpret it, whether it is there or not. I am not interested what is the CO value or what is NH stretching or what is CH stretching. I am not interested. I will ask for only one thing. What is that? Is there any interaction is there or not? That's all. This final conclusion I want. And students will write also. So learn from practice and experience. What is more important is experience is very, very important. That is why still experienced people are respected in the society. Important. The more you practice, the more you will become more expert, and you should have these particular skills in terms of interpretation. Techniques of interpretation, how you understand first of all. Reasonable explanation for relationship should be offered. This is very important. Any data you got, you have changed independent variables, some response you have measured, what is the relationship? Results, mean figures, you will have numbers only. What is the surface? So what is the surface tension? If somebody asks, you will say 72 dynes per centimeter. All are figures. What is octanol water partition coefficient of 2? You will say log. Non-log means it is the figure will be different. So all these, any chemical you take, it is always by number. Aspirin, what is molecular weight number? For water, molecular weight is one number, IR spectra is numbers, UV is numbers, water absorption is a number, everything is a number only. But what is more important is the relationship. This is where exactly you should start with the correlation. In B.Pharm final year, second semester, research methodology and statistics chapter is there. First, it starts with correlation, then correlation coefficient, then regression coefficients. Then you have what is called as modeling and simulation. Then you have got hypothesis testing. So all four will come in that particular chapter sequentially. So you have to establish cause and effect. Unless you get a regression, you cannot establish cause and effect. So some of these principles will be useful in research. Independent variables, dependent variables, they come under research. Then hypothesis testing, whether it is, these are two types of hypothesis. If you want to go for statistical hypothesis, null hypothesis valid, null hypothesis not valid. And my hypothesis in pharmaceutics is whether it is linear or not linear, what concentration. This is my hypothesis. And this is pharmacy, it is required when you want to prove statistically significant or not, you require that. But hypothesis testing is everywhere it is there. If there is no hypothesis testing, you cannot say that all acids will be absorbed in the gastric region. You cannot say that because that is the hypothesis. After doing experiments, of course, that is true. Pick up the thread of uniformity. That is where, one PhD thesis you take. He will do hypertension controlled release, drug number 1, drug number 2, drug number 3. 3 drugs he will do and complete. He will not do one part hypertension, another part diabetes, and third part cardiac like that. He will not do. Same hypothesis is required for him. Uniformity has to be maintained. He will take one drug, 3 methods, uniformity. Same method, 3 drugs, uniformity. Same excipient formulation, different drugs, uniformity. So this is what is. Pick up the thread of uniformity. Today, we have come up to what is called as platform technologies. What is platform technologies? It is the thread of uniformity. They are templates. Just using a template, you change the drug, it will automatically come. It is difficult because research findings are diversified. The thread of getting uniformity is very, very important, but it is very difficult to identify because of multifaceted factors involved in the interpretation. Again, it will continue. Reasonable explanation for relationship. Generalization and conceptualization is developed. So ultimate use of research is to get generalizations, not to get data or tables, concepts and generalizations. Finally, what is your conclusion in terms of generalization? This is one. Even if you say study designs, you say what is called as DOE, optimization, etc. Ultimately, what is more important is generalizations and concepts. The concept of design space is a generalization. If you move in that particular design, you can waive off SUPAC guidelines. And your drug somehow make it class 1 BCS, then you can waive off bioequivalence studies. Surrogate dissolution is a surrogate for bioequivalence studies. Again, this is. So when generalizations comes, I will call two examples. One is BCS class. You know, GL Amidon is the person who has proposed BCS classification.
Okay.
Number one, he is good in solubility. He is also good in solubility. I have seen him for last 30 years. When initially in 1982, when my research work is being done, one of my research paper has been evaluated by GL Amidon on solubility. So that part, this person, actually what the story is, he has taken leave for 6 months. I don't know whether it is true or not, but I heard. 6 months every day he used to go to US FDA office. Sit there. US FDA office is a wealth. All ANDAs will be there. All INDAs will be there. All NDAs will be there. All books, files, all e-filing, etc. So you take any file, complete data is available. Without that, they will not give issue. So that is a bank. He spent 6 years in that writing aspirin. What is solubility? What is permeability? Next, ibuprofen, solubility, permeability. Like that, hundreds of drugs. Just writing tabular columns only. It is not experimental work. Then he tried to understand what is the commonalities in solubility and commonalities in case of what is called permeability. Based on these particular figures of hundreds of compounds, he has developed a knowledge bank. What is the outcome of that knowledge is BCS classification. Now with BCS classification is the first one only. It has started with the GL Amidon. And today, every B Pharm second year student knows BCS classification. And it is only hardly 10 years old. See how much impact. After BCS classification, BC classification, drug disposition also came. And recently, TCS has come. Transdermal Drug Classification System. TDS, TCS classification system, transdermal, like or software company, TCS. So therefore, one classification, this is all called as what is called as a knowledge-based research. You look at this Lipinski rule. Lipinski rule is known as RO5, rule of 5. This 5 means not 5 rules. Each rule will have a 5 number, that's all. That is why it is called as rule of 5. The molecular weight should not be more than 500. The partition coefficient should not be more than 5. The hydrogen donating groups should be 5 minimum. See, hydrogen acceptor groups, 5 or multiples of 5. Everywhere 5 will come. This is the interpretation. And when Lipinski is the scientist who has done this, actually, in fact, I had an opportunity to read his original article and experimental. Excellent work. Today, you have so many of these rules are available. In artificial intelligence, they are called as knowledge-based systems. Based on this, knowledge-based systems are available. So above are called as knowledge-based interpretation. Suppose, say for example, INH. INH isoniazid is lying on the chemistry cupboards for tens of years until the TB target site has been identified properly. Then they have taken isoniazid, has become a drug. But chemists knows isoniazid is a chemical. This is because of underlying process is found. Reasonable explanation for relationship. So like this, if you see the history of these drugs and discoveries and all those, you get several important contributions. All these are because of interpretation. Extraneous information is also important. Several times extraneous information we will set aside, but this is important because somewhere there may be some relationship. Today you may not be able to explain, but it should be properly recorded. Maybe your junior may be able to offer an explanation, or your teacher may be able to offer an explanation after 2 years. So it is often a key factor for identifying the problem. Extraneous factors may be age, sex, weight, etc. in clinical and paraclinical experimentation. Outlier should be properly explained. This is very important because already US FDA says that you have to take a control 5. In that control 5, control 6 animals minimum required. In that one is outlier, another is dropout. Finally, how many are there? What you can do with this? They have to be properly explained because a new hypothesis will come. Because all potent drugs will not follow the known pathways. This is a simple yardstick. Several examples are there. If drug is potent, its pathway is altogether different. You can't expect. If this drug is an outlier, this patient is exclusively a different person. But you have to identify whether it is phenotype or genotype. So exceptions to the hypothesis must be explained. Again, in the interpretation, techniques of interpretation. Dropouts should be proper. Solubility and crystallization, penicillin. Penicillin would have never become a drug if Alexander Fleming has not seen the extraneous aspects of it. Like that, several instances are there. This is like, in fact, after sulfonamides and dapsone, there is no antibacterial drug for 40 years. Today, after that, the only antibacterial drug is norfloxacin. Floxacins are the next generation of antibacterials. After sulfonamides, etc., only antibiotics ruled the world. And nobody could get real antibacterial, that means chemical antibacterial, for a long time. Particularly this one, fluoroquinolones, that is DNA gyrase inhibition, is tremendous. Consult a person who has insights in the study. If you are not able to do interpretation, at least ask somebody, ask some people, different people. Somebody may be able to guide you or help you or interpret you in a proper manner. This always happens with most of the scientists. In fact, I also happened to see several times these type of, they will keep running because that inquisitiveness will be there. They will have data, they will come. And sir, can you help me this? Okay, let us sit and do something with this particular one because data is there. Data is value. Data you can't lose. So when confronting with new knowledge particularly, see, everybody knows flavonoids, but biflavonoids, 2 flavonoids are joined together. Very tough for identification. Across the India, totally only one person we have seen. This person is a chemistry professor in Delhi University. From Jammu, we traveled to Delhi University, catch hold of him, put the spectra on his table forcibly. He is an expert. We know that flavonoids expert. So he saw this structure, spectra. Immediately he got interested. He spent 4 days on that only. Just got interest. So something you focus in, somebody is there to help you. There is always no problem. One person or other will help you. For research, everybody is favorable. So enhance utility of research results. Maximum results. For small effort you do in the laboratory, information should be more. This is almost like DOE type of philosophy. Foreign articles, acknowledge, referee notes, etc. These are to be consulted. Even now, if you have a problem with the interpretation, you write to that author, he will help you out. There is no problem. At least I have seen a few teachers who writes to the author and get the clarification, who send the data and ask him to interpret. Any type of academic doubts, etc. If you express, contact those people, they will be there to help you. And of course, today now you have internet, that research gate, something like that is there. You post your question, they will give you answers. All across the world, everybody will give answers. Of course, whether it is feasible or right or wrong, you have to interpret. But there are people to help you. There is no doubt about it. You just write your question that people will keep on answering. This is a new age of the computers and all those things. So therefore, consult a person who is having insights. Normally, we normally hesitate to consult people because we don't have faith on our own data. What other person will take, you get exposed. So many type of inhibitions will be there. This is because of here complacency in our compromise in doing the research and all those type of activities. Anyway, let us not talk more about it. Consider all relevant factors is another technique for interpretation. Avoid false generalizations. This is very important because any researcher, he will just go by false generalization. Immediately he will go for generalization. But generalization should be done after considering all the points. Hurriedness should not be there. Knee-jerk type of movement should not be there. Because somebody asked you something, immediately you give. No, you give next day. What is the problem? Some question had come, you don't answer them immediately. The next day you answer, or after 2 days you answer. Where is the hurry? Question is given. You try to understand. If it is ready, you give. Otherwise, there is no hurriedness. Hurriedness should not be there. Sir, tomorrow is the last date for submission of the thesis. I have to complete 20 pages and 30 graphs and 40 equations. Nothing can come out from that. So the entire interpretation will go. You write something, cook up something and put it there on the table to get M.Pharm degree. Hurriedness must be avoided. This is very important. Even for PhD scholars also, it is very, very important. And most of my students, Aparna, also knows this. Hurriedness will never work. It will take time. Initial conclusions may always turn wrong. What you assumed will be always, this is a statistical philosophy. What Taguchi says is, the first product you prepare will be always an inferior. And that product you will show to FDA and get the approval. The attender or technician who works in the factory after doing several batches, etc., that but latter period product is superior, not the first product. Simple example is a Day-1. Day-1 came into the market as a drug. But afterwards, Day-1 is not the actual drug. Oxazepam is the actual. The first metabolite, oxazepam, is the one that. So even if you take Day-1, it will be metabolized into oxazepam, and then it will give you the required therapeutic activity. So what you are telling, the first drug, go to the FDA and approve, but it is always like. And you have a large number of examples. First metabolite is active, second metabolite is active, that metabolite is having a prolonged action, drug is not having a prolonged action. So several prodrugs have come into picture. and drug metabolites have become themselves drugs. Chlorpheniramine maleate, dextrochlorpheniramine maleate, ejecta, isomer. So like that, the first conclusion or generalization, whatever you do, you have submitted a thesis today, and after 5 years, you open your thesis and see, is it really correct or not? Then you will yourself can judge whether it is correct or not. That is interpret. So therefore, never go into false generalizations. You should have a clear, thorough, of course, thorough in terms of mind, in terms of data, and also in terms of literature search in the library. These three are required. So these are the techniques. Analytical is very, very important. I don't know what is the time. I will take quickly 5-10 minutes and yes. So analytical is very important. I will tell you one example. I will not read all these things. One example is take dissolution. You have got dissolution basket, the readings are taken, and you have drawn the graph. Tell me what the graph tells. What the graph tells. It will give a curve, that's all. What is the meaning? You have to model it. What you will do, you will first test whether it is first order or zero order. If you don't do first order or zero order, your resolution data has no meaning at all. What the graph will tell, what the data will tell, you have to do that. If it is first order, it is an immediate release product. If it is a zero order, it is a controlled release product. So you have to do that. Simply having data, you just fit it into the Iguchi equation, whether it is diffusion controlled or dissolution controlled, Pixel-Crowell cube root law, or Course-Meyer-Papas equation, swelling studies. You have to model it. Without modeling, you cannot do it. You have to model it, you have to simulate it, then only the results will work. So interpretation also includes modeling and simulation. Without that, it is very difficult. At this particular context, I will tell you one more example. Say, for example, ibuprofen pharmacopoeia says dissolution you should do in alkaline media, 6.4, let us say. Is it correct? No, it is not correct. Ibuprofen will be absorbed from the gastric region. So it should be done, dissolution should be done only pH 1, not 6.8. Is pharmacopoeia wrong? Pharmacopoeia right? Because pharmacopoeia wants dissolution test to be done quickly, within 45 minutes and get the result. That is the objective of pharmacopoeia. But your objective is research. Because pharmacopoeia has given that you can't do and submit your thesis. You have to do pH 1 also you have to do, 4.2 also you have to do, 7 also you have to do. You have to completely mimic the evidence. Then only you will get correct interpretation. So what pharmacopoeia gives, you are doing the same. These things will not help. I will give you one other classical example where students will commit mistakes. Everybody knows that before 220 nanometers in UV, there will be cutoff effect, solvent cutoff. Several people report in the literature that they have done the analysis at 221. Is it reliable? Do you have backup proof to say that it is reliable? It is solvent cutoff. If you are right, you prove it. Otherwise, you just avoid it. Because it goes into the false interpretation. You never know where exactly, what exactly will happen. Okay, so I just gave you, share some of my examples where it says analytical modeling is very important. Even design space is also a modeling. Even factor space is a modeling. So with that is why DOE has become, and that has become one full unit in 4th year, 2nd semester statistics and applied research methodology topic. Development of corollaries, make amendments and modifications. Common factors should be identified. Kinetics are zero order, first order, Higuchi equation, all those things I have explained. This is only just writing steps. Data must be appropriate and adequate. That is why most of the times USFDA gives the how many samples you have to take. There is a separate chapter on samples, how to take samples. Say for example, when you want to weigh tablets, you have to weigh 20 tablets and take average weight. Why 20 tablets? Why not 10 tablets? Why not 40 tablets? Because if you say that if you have a turret, that is tablet machine, 16 stations, in one round, 16 tablets will come. So 16 stations, if you take, even one station is wrong, you will identify what is the problem with that particular upper punch or lower punch or die. So you practically, your sample size should be able to test each one, though you apply statistics. Even dissolution also. If you fail in first 6 tablets, take another 6 tablets and do it. If you fail there also, you take another 6 tablets and do it. N is equal to 20, N minus 1. And of course, N value statistically now has been changed with the process analytical controls, where online testing is there. Online testing will be done more number of times. So now all statistics have been changed N minus 1 to N. Anyway, that is a different aspect. What is more important is data must be appropriate, adequate, trustworthy, homogeneous, apply statistics, ANOVA and all these things. And dimensionless, this is all engineering aspects that are the same. Cautious with errors, because errors should be well controlled. Too much extrapolation is not good. Wrong use of statistics is again goes wrong. Interpretation, validation, p-values, again there is a problem. Interpretation of intertwined analysis, computational checks, etc. All these things, validation and comparisons. Identify and disengage factors that initially hidden to the eye. So constant interaction with the hypothesis is important. So these are some of the aspects. I think I have nearly reached that. I will stop here. I will share this PPT with Padmalata madam, and you can take this PPT and you can use it also for your classwork or anywhere you want. Or you want to give one more lecture to another person's on interpretation, you can use this material. There is nothing like copyright. All students you try to use, try to get benefit from this and also propagate for new generations. Padmalata madam, for giving me an opportunity to share some of my thoughts with you. I think this is okay, 1.5 hours continuously taking, because all of them are highly motivated listeners. So I think it will be useful for them.
Sir, yeah, we should thank you, sir. We are very much thankful for you. Thank you for your rendering our students so much of knowledge, so much of inputs on data interpretation.
Thank you.
I can just tell that you are a polymath, a very knowledgeable person, and we are very fortunate enough to interact with you, to learn from you today some of the aspects of data interpretation. Thank you very much, sir. Now the session is open for discussion, and students and faculty members, if you have any queries, if you want to share anything.
Anything, yes.
Yes. You're right, yes. And sir, Dr. Aparna is here with us.
Yeah.
She wants to interact with you, sir.
Right. Last year I have visited her house and she took me on pilgrimage to different etc.
Okay.
Sir, good afternoon, sir.
Good afternoon. How are you, Aparna?
I'm fine, sir. I'm fine, sir. How about you and madam, sir?
Madam is okay. We are okay. We are able to completely maintain our health. First wave, second.
Yes, sir.
Try to keeping up well.
So nice to hear this, sir. And
Nice to hear.
got a A very good opportunity to hear you after a very, very long time, sir.
In fact, your transdermal work, research, PhD work, I have used in writing a chapter in DOE.
Oh, sir. Yes, sir. Yes, sir.
That I have used that also. I have seen.
Yes, sir.
Some of these PhD works I will translate for the students, for younger generation, for calculations, etc.
Very nice of you, sir. It will be very helpful for all the students, sir.
I can add that it not only helps the students but also faculty members and
Yes,
Yeah, yeah, right,
the
right.
new upcoming generation.
Yes, 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 that is true.
Yes, sir. That interpretation when you were explaining, I was telling the same thing. Like we have done a lot of designing of expectation and all that we have used, and the interpretation you have
Yes,
given
yes,
me,
yes,
everything.
yes.
I was just recollecting, sir. Just
Right, right, right.
very nice
Good, good.
having you today in the college, sir.
Yeah, yeah.
Yes, sir.
Okay, Padmalata madam, any questions are there? Staff members, let them ask anything. I am happy to listen to the questions. So that
Students,
found it late.
students, I request you to post your questions if you are hesitant to ask
Yeah.
post your questions in the chat box.
Yes, yes, it is 1 o'clock. They are also must be hungry.
Nothing like that, sir. There is one more session after this.
Okay,
We are going to
okay.
continue the session, sir.
Continuation.
Okay, yes, sir. Students, one more thing I would just like to introduce to you all about, sir, that is whatever textbooks you are reading, you are studying in your B.Pharm on the pharmaceutical engineering and physical pharmacy and biopharmaceutics, these are all written by sir, whose talk you have listened to you just now. How the textbooks have been helping you in your classroom, you know very well. How simplified the versions are, how intelligible the language is to you all. Somebody wants to speak up, I think.
Okay. Okay, madam, then
Okay.
we will wind up. Okay.
Okay, sir. Fine, sir. Thank you very much, sir.
Welcome, madam. So nice of you for inviting me, listening to you.
Thank you, sir.
Any questions?
I think we don't have any questions, sir.
So I will take leave and go to college, madam, afternoon.
Okay, so fine, sir. Fine, sir. You
But
may leave the
tomorrow
session.
probably I am interested in those 2 lectures. Probably I may join.
Sure, sir. You're welcome, sir.
Data
Please send the
dynamics.
link once again, sir.
Same if you have another share with
Same
me.
link. Same link.
Yeah, yeah. Thank you very much, madam,
Thank you, sir.
for patient hearing. Okay, madam.
Thank you, sir.
That leave the meeting, right?
Yes, sir. There is nothing like it. There you have to fill the feedback form, sir. In fact, to get your certificate, Mr. Devajyoti Vishwasi has a query, and tomorrow the feedback link will be posted here in this chat box. You can fill the feedback form to get your certificate, sir. And yes, we will move on to the next session, that is by Dr. Prasanna from Ayurveda College, Vijayawada. We will just wait for madam to join us.
Good afternoon, ma'am.
Good afternoon, ma'am. You are here?
Yeah, I'm here.
Thank you, ma'am. Thank you.
Can you hear me, ma'am?
Yes, yes, ma'am.
Okay, thank you. Thank you for welcoming.
Thank you, ma'am. And now I would like to request Dr. Praveen to introduce our resource person's profile to the delegates. Over to Dr. Praveen.
Good afternoon, everyone. So all might be exhausted for this continuous sessions. So in this context, I would like to introduce the next guest for the webinar, Dr. B. Prasanna Madam, who is currently working as assistant professor in Dravyaguna department at Dr. NRS Government Ayurvedic College located in Vijayawada. Madam has done her BAMS from the same college and further pursued MD in Dravyaguna at Dr. BRKR Government Ayurvedic Medical College in Hyderabad. And Madam currently pursuing her PhD from Dr. NTR Health University, Vijayawada. So Madam has various accomplishments to her kitty. So Madam has served as member as for the Syllabus Expert Committee CCIM for UG Dravyaguna. Madam also worked as coordinator, coordinator and resource person for the training of Ayurveda medical officers for the government of Andhra Pradesh. Madam also worked as coordinator and resource person for the training of, okay, sorry, it's repeated. So Madam has worked as a member for technical committee for establishment of herbal gardens and wellness centers for the government of Andhra Pradesh. Madam also worked as nodal officer for All India Survey of Higher Education. Also, Madam served as co-editor of Ayush Vignan Health Bulletin and Journal, an official publication of Ayush Department for the government of Andhra Pradesh. Madam also worked as a web organizer of Dr. NRS Government Ayurvedic College, Vijayawada. And Madam has guided 80 forest beat officers for the identification of medical plants and lecture on medical plants, their commercial values in the Annamalai forest, Srisailam. So Madam has also worked as educational tour guide to various UG students and PG scholars to all India tours as per their curriculums. Madam served as an internal and external examiner in Dravyaguna for BAMS students and also in medical statistics for PG scholars. In view with this brief introduction, I will welcome Dr. B. Prasanna Madam to give her talk on recent advances in the Ayurveda research. We welcome you, Madam. Over to you.
Thank you, sir. Thank you for that great welcome. So let's get started. I will share the PPT. Just give me a 1 second.
1 second, Madam. I will make you the co-host. Now you can share the PPT.
I hope you can.
Yes, madam. We can see, madam. Yes.
Yes, thank you. Thank you, Praveen sir. So, in this present session, we are going to talk about, and I would like to share some basic knowledge and in the field of Ayurvedic research and its recent advances. So you might be familiar with the word research, but you may be not as much as familiar with this word with the another term Ayurveda. So let us explore that kind of detailed knowledge in the research in Ayurveda. So here are the contents I would like to share about this research in Ayurveda. So one by one, we will discuss in detail about this, right? So you all know, so the research is comprising of two words, re and search. So that is about the finding out what actually the quest for knowledge through a systematic and a scientific way on specified topic, which is aiming at discovery and interpretation of the new knowledge. So we are verifying the old information or the knowledge of the ones with the application of the scientific methods to get and to know about the new facts. So this is the basic research. So let us talk about what is Ayurveda. So generally in common public, what they have the basic knowledge of Ayurveda is common remedies, simple remedies like a cookbook type of approach. So that is what the general public think about that medicinal plants and their approach, the usage of the medicinal plants in a remedying manner. But that is not the complete Ayurveda. This is not even comprising a 1/4 or 1/5 part of Ayurveda. So this is not merely a conventional science, but it is more much about a conceptual science. Okay, so now talking about this Ayurveda. So as we Ayuhu Vedayatiti Ayurveda. So it is a Veda. It is one of the Upanga Veda of Chaturvedas. So we all know there are Chaturvedas, among them the Atharvana Veda. So this Ayurveda is Upanga Veda of Atharvana Veda. So Veda means why we are talking about these Vedas. So Veda means that is the term determining or giving a stands for authenticity. The Vedas means the word that is whatever that is dealt in Vedas, these are authenticated. That stands for authenticity. So that is why we are knowing and studying as a scientific knowledge. So about this Ayurveda. Ayuhu Vedayatiti Ayurveda. Whatever that authenticated about life. We are all talking about this Ayurveda. What is the authenticated mat is about life. It is a life science. So what is the purpose of this Ayurveda as of today? So present scenario, it has brought some urge to know about this Ayurveda. In this pandemic situation also, each and everyone is looking at this Ayurveda. So the natural calamity of the situation, the present scenario has created an urge, and for that urge, everyone is looking into this Ayurveda. And so what it is actually, the purpose of this Ayurveda or knowing or the living the style as it is mentioned in Ayurveda, the science, it is so in the common way, those people are thinking that is a traditional medicine. Okay, that is somehow it is correct, but the traditional medicine which is can be approached and applied in a scientific manner, right? It is a scientific. So traditional medicine and not only a complementary alternative medicine alongside, it is a scientific. It is absolutely scientific. So how we can say this is scientific? Because whatever the concepts or whatever the amenities that is available in this Ayurveda, whatever that dealing with this Ayurveda is absolutely can be proven on evidence base. So let's see what is the purpose Ayurveda present. What is the purpose of this Ayurveda? So it is not only dealing with this treating the patients, but the main concept lies in the preserving the health of a healthy person. So maintaining the health is more important, the priority given if there are two facets, the maintaining the positive health and treating the or curing the disease. If these are the two facets in the medicine or any science, a medicinal science, so the major role has been played, and the important role was played in the promotive and the preventive aspect rather than curative. So this is the main purpose. Ayurvedasya tantrasya prayojanam swastasya swastharakshanam autarasya vikara prashanam. That means to maintain the positive health of a healthy person as well as to treat the diseased person. These are the two main phases, and that is our purpose of Ayurveda. So now, as we know, come to know about this is Ayurveda. So what are its scope? What is the scope of this research in Ayurveda? So there is a great scope, especially it has a scope in the following themes. So the first and foremost, that is the theoretical research. So in this theoretical research work, so one thing that is about uniqueness in the concepts, and these concepts are, there are several concepts, right, from the Tridosha theory or Panchamahabhuta theory. These are the basic principles of Ayurveda. Apart from a daily applicable and day-to-day life applicable, approachable, and concepts are like daily regimen, that is what we call dinacharya, and seasonal regimen, that is ritucharya, and a diet regimen, that is so these are the basic uniqueness of the concepts. So not just simply treating with medicines. And also the scope in the theoretical aspect is what I am expressing you, that is in the Sanskrit terms. If you have clearly observed, I am giving the slokas. So in the Sanskrit terminology. So this is the area where we can give a research point, research aspect in the translation of the classical text. Nobody, a common man cannot read these Vedas and he cannot able to understand the Vedas. So there is a large scope and translating in the multilingual text or text and books, so that even a common layman can easily understand and apply that knowledge into his lifestyle. So this is the theoretical aspect where we have got this research scope. And now coming to this second one, experimental research. So this is where we generally go in detail about these experiments. So each and every research will be proven and evidence based by the simple experiments, by doing the experiments. So there are various testing modes. So don't think that Ayurveda has never dealt about research. It from the ancient, the ages back itself, they has given a beautiful expression and meaning of research. So I will give you one single word, one single stranger, so that you can understand how this Ayurveda has been dealing with research. So the research is a concept, theory, a thesis or a theory that is created. So this theory has been created, as we can call it as Siddhanta. We are establishing a concept, right? So this concept is Siddhanto Nama Yaha Sa. So this is called Siddhanta. What is called Siddhanta? Parikshakaihi Bahuvidam Pariksha. So the researcher, the Parikshakaha is here what we can term as researcher. This researcher, Bahuvidam Pariksha, he has to do the experimentation and the observations several times in the multi-direction and multifocal. So those pariksha, Bahuvidam, all the experimentations, Hetu Sadhaitva. That means we have to explore the factors which are causing the cause and effect relationship, which are dealing with the cause and effect relationship. Hetu Sadhaitva. We need to what we can say. So we need to establish those factors which cause this action. So that is what we call cause and effect relationship. So the experimentation is done to establish this cause and effect. Finally, Hetubhisya Sadhyatva Stapyate Sa Nirnayaha. So finally, they will come to a conclusion, and this conclusion is nothing but a Siddhanta. That is how we create a concept by doing several experimentations, several observations, and finding out the reasons and the causes and the effect, the relationship with the effect and the actions, so that the research will be fulfilled. And this is how they have beautifully explained in Ayurveda what is really meant by research. And not only that, they have also given explanation of what are the qualities and the abilities of a researcher, those who are participating in the research. So the qualities of a researcher is also given, that is like 6 characteristics. The qualities of a researcher, the one who is undergoing the research should have these qualities. And I'll give the stanza again. Vidya, Vitarka, Vijnana, Smriti, Tatparata, Kriya. Yasya yete tasya asadhyam iti nivartate. So there is nothing you can do. That means you cannot, you never expect it is not, cannot possible. So everything is possible if the researcher is having these kind of characteristics. How is this possible to conduct a research? Vidya, that is a study or a science. Vitarka, that is a logical thinking and a reasoning. Vijnana, the scientific knowledge and the methods to approach this scientific way. Smriti is also the important thing, the recollection and the memorizing the things. Tatparata. So this tatparata is the repeated observations or the repeated experimentations. Kriya. Kriya is what we can say as practical applications. Applicability. So if these 6 characteristics are followed to conduct a research and the 6 abilities are shown by the researcher, and nothing is possible. So everything is possible, that is, you can do whatever you want to do as per the, to explore something as per the term as research. So this is about the, so now coming about the clinical research. The third point is about the area or the factor, that is the theme of this scope for research is clinical research. This is some, as I said, so the preventive aspect and the curative aspect. In this curative aspect, this is the major part, that is about the clinical research. So these clinical research is also comes under 3 aspects. Okay, so that is, one is curative aspect, another one is promotive aspect, and the third one is preventive aspect. So curative aspect is as simple as treating a disease and without developing. So this is very, the half of the part is very important. Treating a disease is not a big deal. So as in the case of a more conventional medicine, other sciences, so the treating a disease is very simple, but with treating disease without causing any complications, without causing any side effects and further developing any kind of adverse reactions. So that is the uniqueness of the curative aspect in this Ayurveda. Next one is the second clinical aspect, that is about the promotive aspect. So promoting the and enhancing the immunity. So this is the basic right now what we exactly need in this scenario, in this present pandemic situation, that we need to enhance this immunity and the resistance power rather than controlling the disease or treating the disease. So this is promotive aspect, and the third one is preventive aspect. So the maintenance of the health. So what the main aspect, that is swastharakshanam, that is the, it is giving the preventive aspect and is giving so many areas connected with that, like the maintenance of yoga, pranayama, dhyana, and it is also based upon some other scope for other sciences which are interrelated and connected with this Ayurveda, that is clinical pharmacology and reverse pharmacology and pharmacoepidemiology. So epidemiological diseases, these are the other areas and the disciplines, sub-disciplines where we can give a scope for research in Ayurveda. So the next point is about need. What is the need for this Ayurveda? Need for research in Ayurveda. Do we really need this? So exactly. So here there are 3 main aspects in the necessity of doing research in this. One is educational, pharmaceutical, and third one is again clinical and practical. So what is the educational aspect? So here in this academic aspect, the globalization. So here we have got this globalization of Ayurveda, and in this we need to explore the historical aspects, whatever that dealt in Ayurveda. And we need to require some separate and Ayurvedic research methodology. So whatever the research methodology and the protocol that is followed by the other systems of medicine may not be applicable for in this Ayurveda. So we need to have a separate research methodology that is about the academic aspect, and also the guidelines, separate guidelines. So as per the WHO guidelines, the parameters and the protocol of this Ayurveda may not be applicable for the present research aspect. So that means we need to explore more about this research aspect in this field of Ayurveda. And the revalidation of the facts. So whatever that said, authenticity, authentication in Vedas. So those maybe they have authentic, but we need to revalidate. So what is the present day as on today, how it is being applicable and validated. So that revalidation of the facts which are enumerated in the Ayurveda, that has to be also explored. And the knowledge has to be grabbed. So this is about the educational angle and the pharmaceutical. So this is the a great demand and the trade market value for these pharmaceuticals, especially when it is about under the heading of herbs. The highest turnover in this entire market, in the trade market, that is about the pharmaceuticals, and that especially about the herbal industry. So if you have this basic idea of this medicinal plants and herbs in the uses in these pharmaceutical companies, the pharmacy, so you might be knowing about its value and this export and import values. So on this trade market, highest trade market, so in this we need to develop some evidence of efficacy. Not just like that, as it is said and done, it is proven, but we have to develop an evidence-based medicine. And what is the efficacy of those drugs? Not only the products, the Ayurvedic medicinal products, it is also about to develop assurance to give the efficacy in the practices. That means not so. I told you that is medicine is the only 1/4 or less than 1/4 part of Ayurveda. So apart from that, that is the therapeutics, that is the procedure, the practices we do like Panchkarma, Kshar Sutra, Agni Karma, leech therapy. So there are number of Sastra, Kshara, Agni. So these are the n number of procedures we follow apart from medicine. So these practices, all these practices also should be revalidated and export. So that is about the, and that means it should be standardized with the quality control of both the products as well as the practice of clinical practice. So next, to evaluate the effectiveness of various formulations. So what, so not just like in this like a tablet form, a capsule form, like an injection. So there are, we have sattva, basma, rasakriya, taila, bruta. So there are multiple types of formulations and the medicine preparations. All these formulations, so which one will be better, like giving in the form of a decoction or giving in the form of a tablet form? So the route of administration and the mode of administration, and so this is and the formulation of the drug, these are also will affect ultimately the disease control. So this is to evaluate the effectiveness of various formulations, and like Nasa Bindu, so in this present medicine as a nozzle drops. So each and every type of method of formulation that has to be done as a research in this pharmaceutical aspect. Now coming to the third, the practical and clinical. So there are some better treatment modalities in order to improve the quality and the therapeutical. Hello? It causing some disturbance. Can you keep it mute? Hello? Yeah, thank you. So here, in the practical aspect, so to upgrade this Ayurveda as a time being, so as per the time to time, it has to be metamorphosed or modified so that the public and the people can utilize and take the benefits of it. So this also comes under this practical approach. So in the previous days back and ages back, when we are using Panchkarma procedures, we need to take some pottery preparations like Nadi Sweda. For example, you take Nadi Sweda, we need to take some, instead of that, as of today, we are using some rubber pipes. So those days they are taking some natural products as the necessaries to conduct so many practices. So as the time being and the as a trendy, we need to modify and upgrade these techniques and tools. That is what we are practicing as of today. So in view of these new diseases and emergency management of diseases, so this is the emergency situation. So in such a condition, what are the modalities what we are taking as a practice mode aspect? So that has to be given an approach of research. So this is the practical aspect, that is the to keep in view of the emergency management of the disease condition. So I'm not only that, so this has got the present um, the worldwide, it has been created some urge and search for this need into as because of too much of mislead training in this ancient science. And if you can see in the multi social media and the public, so many postings, if you can see, there are so many facts. That means unprofessional and unqualified person who are handling this science will also causing some bad reputation to this science. So too much of preparation of like preferences of the cookbook approach. So generally people, what do they think? Those people are more talkative and they have so many communication skills. That doesn't mean they are qualified, right? So without that, unprofessional people, they are taking the advantage of Ayurveda and they are misleading the original context and the science of this treasured science. And too much of business interest, this is the most threatening, this is one of the threatening point. So almost all, everyone is, as there is a demand of one particular herb, that means there is a great business regarding this. So this is it has become a trendy as a Ayurveda as a business formula. So any medicine that has been taken a patent right, they will give Ayurvedic proprietary medicine. The simple tagline, they will take Ayurvedic proprietary medicine. So the public will demand and the public will take it easily without thinking about whether it is genuinely Ayurvedic product or it is really possessing those herbs. And it is, so this is the where we need to go for this research in this. And there are some other aspects, like I have already mentioned, this increased demand for this evidence-based Ayurveda and industrialization of this Ayurvedic drug sector, and the inventing new classical formulations. And I will give you one of the examples for this. So you might have heard about this Tipatiga, right? Tinospora cordifolia, Tipatiga. So apart from this churna or swarasa, that means the fresh juice or a fine powder. So rather than that, we will prepare a sattva. So this is a different formulation. So only Ayurveda scholars can do this such kind of formulation. So it is not available in the market. So like that, inventing the new formulations so that it will be having some instantaneous and a nanoparticle science of treatment and the drug approach, drug delivery system. So this is about the other areas of necessity. And regarding the safety of the drug standardization and quality control assurance, and to develop new drugs and new formulations. That is, so as per Ayurveda, na dravyam na anaushadhi bhutam dravyam upalabhyate. Na anaushadhi bhutam. That means there is no material or matter or no plant in this world which cannot be having or which is not showing any medicinal properties. So that means each and every plant, so that you know about this and number of species and plants. So each and every plant has got its medicinal plant. The only thing you need to explore. So those new drugs and new medicines and new formulations. And you need to explain and improve the understanding of the fundamental principles. So this is very, very important. So nobody ever knows what is the theory of Tridosha, a Tridosha concept. So this is these are the pillars of Ayurveda. So as if I can say, the definition of Ayu, Sattva, Atma, Sarira, Indriya, Sarirendriyo, Samyogo, Dari, Jivitam, Nityagasya, Anubandasya, Ayu, Paryaya, Uchite. The pure definition of Ayu, what is meant by life? So it is not the life sense like expanding or lengthening life. So don't assume that this Ayurveda is only giving the longevity of life, but it is the science which is dealing with this living, the method and the style of living, how we have to. It is nothing to be with the longevity of life. This also gives the quality of living and quality of well-being. That is the real meaning of Ayu. So here the definition, what it is means, Sattva, Atma, Sarira, Indriya. These are the four elements in the Sattva, that is Manas, that is mind. Okay, so this is simple one line is also equivalent to this WHO definition for health. If you can see and explore this detailed manner, Sattva, Atma. So that is about the spirituality, human spirituality, about a mind, that is about a behavioral and psychological manner, the spirituality of the human, and Sarira, that is without body you cannot achieve anything. So that is about the physical aspect, and Indriya, that is about senses. So controlling the senses is the ultimate. So all these four, when they are Samyoga, coordination, Anubandha, cooperation, and Nityagam, they are everlasting. And that means if they are eternal, then only you can achieve Ayu. So Dari, that is withholding. So when they are withholding these four elements, and with the coordination and a combination of these four, and as long as they are being in coordination, as that will cause eternity. So this is about Ayu definition as per Ayurveda. The same which has mentioned in WHO also. The physical, mental, spiritual, and social well-being, that is what generally is called health. So this is about understanding the fundamental principles. So why I have covering this point is to explain and improve the understanding of fundamental principles of Ayurveda in a better manner. So coming to the same need, so among those preventive and promotive aspects, the public is looking forward for the only curative and clinical aspect. So what is the need for this in this clinical research? So revalidation of these facts in Ayurveda classics, whichever that enumerated in the Ayurveda classes. And we need to also establish the doses, the fixation of the dose. So like in this conventional medicine, the fixing, so that is the milligrams and the micrograms, that is not fixed for one particular drug or medicine in a particular disease condition or in a particular individual. This is not any mathematical or statistical thing. So the like as if 2 2s or 4 is universally accepted, but it is not in the case of science. So it not be possible. It varies from person to person, time to time, place to place, individual to individual. So like that. So based upon that, even for the medicine to medicine will also, the doses, the duration and the indication, and with these all the effects will change, will be different. So that should be come to establish, and that should be established in a better understanding. So that is about the clinical research and to find out the new better treatment modalities. So if you are applying Sutra, it will cause some excruciating pain, a gruesome pain when we are applying this thread. So such a way, we need to find some better modalities, treatment modalities for the convenience of the palatability and acceptance of the patient and others. So, and to find out the newer source material like which are whatever the endangered species, we need to substitute them. Which so there are several plants which has been explained in this science. As on today, it is not available. They are almost extinct and endangered. They are in the endangered. So those has to be substituted with some available drugs. So that is also there is a source of need. And to find out the pharmacological basis of mode of action. So right from the entry of the drug and the exit of the drug, so we need to find this. So how it is better. So if we take the so many metals. So general public think it is a heavy metals that should not be having some toxic effects. But we are taking all the purified method, taking some basmas like Vajra basma, Swarna basma, and Rajata basma. So all even the metals are also being made into basmas, and those basmas are being utilized. And how they are acting without causing any damage to the body, and we need to find out such formulas. I'll give you one of the examples. So, like we have given previous we have done a research on this cancerous patient, giving a different formulation that is called perpati. Perpati, that is almost a nanoparticle size product. So this perpati, I mean, which is having a great absorption and assimilation process. So that preparation we have administered through endoscopy. The drug has been administered as endoscopy. So here the different modalities, the different approaches, and right from that in the gastric mucosa, that is the level of absorption there and the site of action. So that kind of pharmacological basis of mode of action and site of action, we need to find out with evidences. So next, under standardizing the treatment procedures, even unscientific person or a professional, unprofessional person will also do the same procedures like panchakarma. Even in the beauty spas, so many people are using this massage therapy, reflexology, but that is not what actually meant to do in Ayurvedic Abhyanga, what we call as Abhyanga, that is a massage therapy. So this massage is not equivalent or no way it is possible to do in the spas, beauty spas, what they are doing in the massage centers or all this. So we need to standardize those treatment procedures also. That is not, we are doing this as same as Ayurveda person, so it is not possible. So, and regulatory approval and registration purposes for evidence-based medicine. So this is about the need for clinical research. And what is the importance of research in Ayurveda? I will speed up because we have very important points left behind. So let me, so please allow me to make it rapid mode. So importance of research in Ayurveda. Basically, this is again also mainly based on 4 factors. One is basic research, applied research, evaluation research, and action research. So basic research to improve the individual quality of life and to increase the human life expectancy. These are the new concept, new conclusions, and establishing the new facts. So, and applied research, the facts could be established only after careful observations and experimentations. So, and evaluation research, Ayurveda is gaining global acceptance. Undoubtedly, it is taking the global acceptance. So, the interest of the public and such a demand only to be fulfilled by developing an appropriate research methodology. The research protocol is very important, and separate research protocol that is important for evaluation of the research, and standardization, quality control for Ayurveda products and practices, and for easy administration and quick and rapid action of the drug. And action research. Research is useful to assess the relative effectiveness in the healthcare programs and health policies. This the government is seeking towards, like as you can see, there are coming Ayurvedic vaccines and injections. So this kind of potentiality in showing the solutions for the human health as trendy and as per the need. So it is a relative effectiveness from the governmental sector or from the private or NGOs, whatever it is. But healthcare programs and policies should be established. That is about it. Now, utility of research in Ayurveda. So our um, Rishi Cheraka, he says that utility of research is those who alone are wise who act after investigations. That means the person who is undergoing so many investigation experiments, only those people are be wise. And so we have this new strategies. So simply we have some testing tools in our Ayurveda, that is called what we call as Chaturveda Pramanas. Okay, Chaturveda Pramanas, and Pramanas means the most techniques, okay, tools. So and the first one is Pratyaksha, that means by direct knowledge, the achieving the knowledge by direct perception of senses. Pratyaksha Jnana. Anumana, by inferential knowledge. And the third one is Aptopadesha, that means whatever dealt earlier in the literature on previous knowledge, that is about scriptural knowledge. And the fourth one is experimental knowledge or the true knowledge, that is what we call yukti, as per your experiences, experiential knowledge. So these are the four testing tools what we take consideration for this evidence and standardization of this. And the strategies for obtaining the new knowledge is the reasoning, experimentation, comparison. This is also important. Upamana. So one is compared with the other. So comparative studies is also one of the major part in the research. So and the judgment of by taking clues from the text, classroom text. So this is the scriptural knowledge. So these are all the strategies and the testing tools in this, the how we are utilizing the research in Ayurveda. So next, the here, though these have this. So when we talk about this SWOT analysis, the strength, weaknesses, opportunities, and the threats. So here the problems and the threats in this are lack of documentation. So everyone is doing, and every year they are almost conducting nearly 2,000 to 5,000 researchers in this Ayurvedic institution. Only the teaching institutions are what I'm talking about. Apart from the research organization, we are not talking about. Only the PG holding college institutions are nearly 2,000 per year, 2,000 to 5,000. So these, as there is so many research findings are there, but they are not being published or documented. This is the problem. Okay, in the global arena, that is about. And present day, other researchers are failing in this respect as they are unable to disseminate the knowledge gained from their research works. So all the research activities are done blindly following the modern trends. This is a very, so that is what is said. Modern protocol and modern research methodology may not be applicable as the testing tools are different for Ayurveda. So we need to set up a separate guidelines for this, strengthening the knowledge of this science and the lack of evidence-based medicine. So these are the problems and threats we have. And there are several questions arising what to do with this research and what are the methods needed to create evidence in Ayurveda, which study designs should it be adopted. It is may not be possible all the time, observation and experimental. So several times we need to do a comparative, a cohort study, case control study. So apart from this randomized control RCTs, so do randomized control trials, clinical trials, randomized. So lead Ayurvedic evidence, how to structure these clinical studies in Ayurveda, and methods empowered to achieve evidence-based Ayurveda. If Ayurveda is really such effective, great effectiveness, then how to show its ability to alter the natural course of disease. That means right from the entry of the path. So and that throughout the pathogenesis. So if you see the different phases of the diseases, at each and every phase, how the drug and how this treatment of Ayurveda is dealing with particular that stage of disease condition. So these are the questions, and what kind of documentation we need to represent and we need to publish. So these are the questions rising as per the research in Ayurveda. And there's some basic worldwide and global problems in the clinical research, that is understanding of fundamental principles and the selection of the subjects. Subject is on whom we have to conduct this research, the subjects selection, because the disease what they term used in modern aspect may not be the same as in the Ayurvedic aspect. Okay, so selection of the subject and the diagnostic approach. We have some parameters, a specific subjective and objective parameters. So for the diagnosis purpose, that is entirely different from this, like threefold examination or observation of the patient, eightfold observation examinations, and tenfold examinations. So these are all the different fundamental principles of examination and the diagnosis of the disease. Like Darsana Sparshana Prashna, that is about the threefold. Like that, we have 3, 8, and 10-fold different factors that is taken into consideration for diagnosing the disease. And use of quality of life parameters as assessment criteria as mentioned in classics. And dependency of researchers on symptomology. This is very the biggest problem here. So what are the subjective parameters we consider in the research only taking as the symptoms? But the symptom is not the only criteria, the maintenance of the health and improving the resistance power. So that also should be considered. And understanding of Ayurveda classical terminologies. This is the biggest problem here. This is a burning problem, the terminology. So generally diabetes mellitus, we all know that diabetes is a blood sugar, increasing blood sugar levels. So generally in Ayurveda terminology, generally people talk about Madhumeha, but so we take it as Prameha and Madhumeha together and Vatajameha. So there are several, the terminologies in this Ayurveda may not be correlated with the terminology exactly in the other common sciences or acceptable conventional science. So this is about the problems facing in the clinical research. So naming the under terminology, naming of the disease itself is a big problem. Right? So and worldwide surge has created some problems, and that problem is demand. Demand for safety, efficacy, and the demand for the new drugs and education practices and pharmacies. So these pharmaceuticals are the main, what you can say, especially with those who are taking these herbs and traditional medicine. We need to have some strict regulations and availability of authentic source of material and exploitation, over exploitation. Non-compliance of good agricultural practice. So this agricultural practice, so if you go to the market and you are taking some, collecting some raw material, so the first and foremost step we face, the problem we face is the adulteration. Adulteration of the drug. You cannot find a genuine drug what you are going to do a research or you are creating a medicine. The finding a genuine drug itself is the main problem. So here we need to find some good agricultural practice, a good cultivation practices, so that the original drug and a genuine drug is available for preparing the herbal medicines. And commercialization, as we all know, so this is the trade of the herbs leading exploitation, adulteration, and trade rivalry also. So one pharmacy will be creating one sort of medicine, the same medicine will be created by another pharmaceutical company. So there will be a tie-up with this, which is better. So like that, a trade rivalry between the pharmaceuticals will also the world creating such kind of problems. Now the solutions. So these solutions in the SWOT analysis we can consider them as opportunities. We have number of opportunities, though they have some problems and weakness also. So here, creating a consensual Ayurvedic research methodology and which is suitable to useful for Ayurveda, and national grading system. So here in this point, Ayurvedic terminology, I would like to mention one point, that is NAV-STP. NAM-STP. So this is NAM-STP. That means the abbreviation of this NAM-STP, National Ayush Morbidity and Standardized Terminologies Portal. this is one of the database portal which has given different like ICD-11. So the preparation of ICD-11 complete Ayurvedic disease classifications. So Ayurvedic disease classification has been created in this NAMSTP, that is National Ayush Morbidity and Standardized Terminologies portal. Okay, so this is one of the solution they are looking for, and it has yet to establish. And the preparation of this Ayurvedic disease classification and institution of yearly awards for the publication of this, because the documentation and publication is a lagging behind. So here we need to encourage and give benefits to institutionally, not individually, institutionally. So they should award something so that they will be encouraging documentation and updation of the research journals. So once this earlier in 1970s, 1980s, the CCRS has given some research articles like MAPA, medicinal and aromatic plant. So this kind of articles now they have changed to research portal, Ayurveda Research Database, ARD, Ayurveda Research Database. And I will give you some of the website links also so that you can go through this. What is the present research that is undergoing? So the CCRS, the abbreviation is Central Council for Research in Ayurveda and Siddha. That was when it was established in 1976. So now it has been changed, and the full form is like Research in Applied Sciences. So almost all the sciences, all the Indian system of medicinal sciences will come under this CCRS. And National Institute of Ayurveda, the Ayush Ministry, we have a separate, established in 2014, we have a separate autonomous ministry.
Hello, madam. Hello.
No?
Hello, ma'am.
Hello. Yeah, can you hear me?
Yeah, now we can hear you, ma'am, but we don't
Oh.
see the slides.
Yeah, I will share it. Sorry for the inconvenience. I think it was a network problem or disconnection was there.
Okay.
Madam, is it visible now?
Yes, ma'am.
Yeah, fine. Thank you. So this, yeah, Ayush Ministry in 2014, there is an autonomous Ayush Ministry was started, and as per them, so each and every research articles are being published with easy and convenience in the Ayush Research Portal, given by the Ayush Ministry. So this is about the solutions what we can find about our documentation and the disease terminologies, and these are the points. And now suggested approaches for creation of better scientific evidence. So here I would like to say one word, that is absence of evidence is not an evidence for absence. So general public think as it is no evidence, that we cannot say that is not existing. Right? Absence of evidence is never it will never be an evidence for absence. So, but even though the word looking for evidences, so that is why, as per the being um, uh, social being, we need to accept this and we need to prove our evidences. So evaluating that objective parameters of basic classical methods, that means a standardization, diagnostic, therapeutic assessment for validation of the traditional scientific medicine or complementary alternative medicine. and evaluating and evolving criteria for prioritizing the research opportunities. And this is the some of the solutions and suggestions for this improvement in this research outcomes. Consistent quality products and the standardized operative procedures for doing any kind of trials, drug trials or clinical trials, and sincere efforts required in all the agencies. We have some of the foundations and agencies in, I'll give you one of the examples for the cultivation of these medicinal plants, giving a subsidy by the government and giving awareness and knowledge to the farmers how to cultivate the medicinal plants, as it is being a great need here. So this, like I'll give you one example, that Kovalam Foundation in the Vizag, Visakhapatnam, with a very less cost, they are giving the seedlings and they are promoting the propagation of this medicinal plants, and such a kind of sincere trials from the government sector and the private sector and the NGO agencies, they have to come forward. So hand in hand, it is not just one person cannot change the entire this systems. So one, we have to be work hand in hand so that we can improve our ancient sciences and our treasured sciences. So next, the complaints of GCP, GAP, GMP, and GEP. So what are these? These are the guidelines for good cultivation practices, right from the cultivation of the medicinal plant, and good agricultural practices. So here, the cultivation also includes collection of the drug. So the method of collection of drug is also. So for example, I will give you aloe vera. If you think aloe vera, so aloe vera has a great, as a pharmaceutical students, pharmacological students, aloin, right? So this aloin has a great value and export value in the world trade, right? So, but when it has to be collected, the right appropriate time, so within 24 without getting oxidation, so that has to be collected, and that aloin is made into fine powder, so that will be effective in the reviewing this as a laxative. So this way, the good collection practices, good agricultural practices, and good manufacturing practices, and along with said good educational experimental practices, So we need to set up some guidelines. So these are the next global research requirements. What are the basic? So I told you the basic requisites of a researcher, the qualities and the characteristics of a researcher. Now the requirements for a research in the global and lifestyle diseases and their prevention and management. So this is a area it is required, and pharmacological researches in the field of known herbs and as well as unknown herbs, and research in the field of panchakarma and yoga. So better standardized procedures and study of local flora. So the government also giving a district-wise available medicinal plants in the tribal area and tribal health care also. Tribal people and those people using these tribal herbs and as a medicine. So that should also be been studied, and study for local flora, for Ayurvedic pharmacodynamics, and research in the field of food technology. If you know, you should come to know about this heritage. This is a heritage company. We are very familiar with this, like dairy products. But now this heritage is looking forward to nutraceuticals, with herbal nutraceuticals. They are making some ashwagandha milk, vitamin milk for ashwagandha. So like that, we are developing the food supplements from the medicinal plants also. So that means food products from the herbs, that is also should be used in Ayurveda. So next, research in the field of agriculture technology to grow. So one of the cultivation methods what we use. So here, Sarpagandha, take example of Sarpagandha or Ashwagandha. So majorly, almost like 17 to 18% of all the medicinal plants, the useful part is root. That means if you are cutting that root, that is absolutely you are sacrificing that plant, isn't it? So what should be done? So we are applying some new techniques like a bamboo cultivation. So that means those roots which can expand like Sugandhipala, that means yeah, Sugandhipala, you are familiar with that, right? So these are very elongated roots and taps, so that you can using different techniques like bamboo technique and the root cutting method. So this in this I'm unable to show this image. So there will be a mesh like part around this taproot system of this plant. Whatever the tertiary and the rootlets, that will be made into a fine blade like structure around this. At first we need to make it, you have to dig in the central portion of this root, and the later roots should be collected without sacrificing the main root. So even for the adventitious roots of the different rooting system also we can collect, so that you are surviving and you are not causing endanger or extinction of this medicinal plants. So such kind of new techniques of the cultivation is also should be approached and applied as a latest technology, and different strategies should be applied. And the areas for classical Ayurveda and that requires further exploration and updation, that is updation of differential diagnosis of the diseases and the development of diagnostic tools in the Ayurveda. I said the testing tools are there. So like that, 3-fold, 8-fold, 10-fold diagnostic tools are. So that also should be developed as per the requirement. And study and finalization of pathogenesis of new diseases. For this so-called general line of treatment. So when there are new diseases are coming, we need to finalize what is the line of treatment for each and every disease that should be formulated. And inclusion of the new herbs. So that is not as possible. So some of the plants that is exotic plants. So that is also to be taken into consideration when we want to create and include some new herbs in this Ayurvedic Materia Medica. So they have enlisted. So each classical book they have enumerated or enlisted 500 plants. So apart from that, whatever the new herbs that should be added into this and included, so that it is studying, and we need to study their further pharmacodynamics and study of new doses form, like a capsule form, either in the injection or vaccine form. So on this pharmacotherapeutics, new Ayurvedic pharmacotherapeutics should be the areas of these requirements. And fundamental principles of Ayurveda while designing the research work. So first of all, we need to consider this physical constitution, and next consideration of the psychological and behavioral science. So consideration of the stages and phases of the diseases, different stages and different phases of the disease, and fixation of the doses. So this fixation of the dose is dependent upon several factors like age, gender, your diagnosis, one's digestive file, a metabolic rate. So these are all taken into consideration while we are fixing the dosage of the any drug. And drug administration time, drug administrative routes, vehicles and adjuvants which will enhance and which will act as a catalyst for boosting the absorption level of and absorption and efficacy of the drug. That is the vehicle and adjuvants, and the diet regimen. So we have a common saying. So as long as you're using medicine, as long as you're following a diet regimen, there is no need for medicine. Okay, so even if you're using medicine, so that is useless when we are not following this patya and apatya diet regimen. So different formulations and various routes of drug administrations. So these are the fundamental principles that we need to consider while we are doing research in Ayurveda. And these are the merits, what we say the strengths. in the SWOT analysis, the strength and merits of this complementary Ayurvedic medicine, that is this culturally it is competent and sensitive and holistic approach. This is very, very important. Holistic is not cruel type of treating the patient as. It is most holistic approach, cost effective, and it is natural and eco-friendly. So this is also one of the important aspects. And minimal adverse effects, easy accessibility, and prevention aspect, preventive aspect, and emphasis on the prevention and health promotion and quality of life. These are the merits of this complementary or alternative medicine or a traditional scientific medicine. Now we are entering into the important core part of this topic. It is about the research activities. So what is the advances? So now we are justifying the term, that is recent advances in this research. So here are the research activities. So we have made a research database and a separate software. So not absolutely available for all the researchers, but it is only available for the intramural research that is being. So here I am presenting you research project EMR and IMR. So EMR, that is extramural research. IMR, intramural research. So this EMR means intramural research. Whatever the research that is being done under the roof of this CCRS and the government sector, central government sector, that is comes under IMR. And the sub-disciplines and the other departments and the different other sciences collaborated with this CCRS and the central council. So those research works comes under EMR. So in this IMR, these fundings, these researchers are having course of funding budgets. Such a large scale projects are being undertaken by this government. So here, so the research that is coming about the Ayurvedic medicine, or what we can say IL-64, and what we have generally recently created this medicine for this COVID-19. So they have done the collaboration with ICMR. So which comes under EMR, okay, extramural research. So absolutely that was the research undertaken by the Ayurvedic, this applied sciences of the Central Council. So their people are using this software, the research database and the software being, it is not being applied and accessible for the rest of the research. It has come to start about to come, and now and bioinformatics. We have bioinformatics centers which are also collaborated with this research activities by the CCRS, and the research articles and publications. So if you go to this website ccrs.nac.in, I have shown this website name also. So there you can see the articles in number of lakhs. Okay, thousands and lakhs of these articles are published, and so that is about the what is the activities that is undergoing in this research in Ayurveda. And health policies and Ayurveda policies and health policies, like even in the Arabic also we have started newly introduced this Ayurvedic treatment, not only Ayurveda, Ayush systems. Like the government has taken several schemes and policies, like we can say Shishu Poshana Abhiyan. Recently that started around 2019, I think. So Shishu Poshana Abhiyan, so at the time of this International Mother's Day, Saptaha or Poshana Mah means month, Paksha, Poshana Abhiyan Paksha, that means Paksha means 15 days. So 7 days program, 15 days program, and a 30 days program has been created especially for the maternal and infantile health and their benefits. Okay, so in the month of this March that will start. So that is one of the scheme that has started as Shishu Abhiyan. And Ayush Samvad. This is the recent policy they have created. Ayush Samvad means So each and every school, government college, government institution should be taken advantage of this awareness and wellness centers, and they will give awareness camps and medicinal camps month-wise and quarterly, every trimester. So regularly they will be in coordination with the academics of the public. So this is the channel with the students and the youngsters. This Ayurveda will be more flourished and worldwide acceptance that is possible by this Ayu Samvad. Vad means discussions and talks about this Ayurveda in the academic institutions. But now it has started only in the government sector. It is about to start in the private colleges also. Then each and every college and the school and high school, even primary schools also, we are giving some basic this wellness, creating awareness about this Ayurveda. That is about Ayush Samvad and Ayush Gram. This is another policy. So here Ayush Gram means, so the rural people, those who are not about to, so generally the PHCs and the CHCs will be one in 3-4 mandals. Okay, that is not possible in each and every village. So the PHCs merely they are established in mandal wise establishment here, the PHCs. So, but there are some sub-centers on all these things. So the primary health centers. So, but as per the Ayush Gram, each and every village there will be Ayush counseling by the Ayush doctors, and that means, so those who are working in this mandal area, those doctors, they will choose one village and they will adopt this Ayush Gram, means adopting that financially and academically, whatever that what we can say, backward people and the backward people, public, and where there are more the dense areas, those being selected as an ashram, and they will develop, and they will develop the sanitation condition, all the health, not only one department, not just the health department, even for the agriculture, veterinary science. So whatever the related with all the sciences, so that Ayush Gram will be adopted, and whatever the public, whoever the public listeners, they will get the benefits of this Ayush Gram. Okay, so all the activities and benefits are being done in this Ayush Gram. That is also one of the policies. So like that, there are any number of policies and schemes being started by the government, especially present in this government, and there are some scholarships and schemes, especially for the PG and PhD scholarships for the research that is done by the Ayurveda scholars, and the different schemes. And here I told you medicinal plant cultivation. In the government sector, as they are promoting this, giving a subsidy for the cultivation of the more medicinal plants, even they will give a connection with without, this is a very important aspect in this, directly whatever the collected outcome of the medicinal product is directly being without any mediator. These are brokers or these persons. So it is directly being given to the pharmaceutical companies. As I said, Covalent Foundation. So like there are each and every forest areas, there are some tribal foundations, and these foundation, what they will do, whatever the collected material and the product, a medicinal product, that and the time being very timely collected, so that is being directly distributed to the pharmaceutical companies. So they will get the full amount and the required amount income as per the income, as per for the quality, and for the, and there will be an Ayurvedic doctor who can, I mean drug inspector, Ayurvedic drug inspector who can verify that genuinity of that drug. So in this way, they are promoting so many medicinal plant cultivation practices and methods. And there are some nurseries also given. So that means some urban areas. So don't think that is only focused on the villages and the rural areas. Even the urban and most concrete of areas. So here they are giving a free distribution of, and they are promoting establishment of nurseries, medicinal plant nurseries, and they are giving at times, they are giving, not all the time, at times as per the possibility they are. So this is done by the NMPB, National Medicinal Plant Board, which is being supporting to the state-wise medicinal plant board, and they are giving funding for the free distribution of the medicinal plants. Occasionally, they will give some paper notification or the advertisements also when they will give this kind of nurseries will promote this kind of free distribution of medicinal plants. This is a kind of a minor projects that is being undertaken by the NMPB. That is a National Medicinal Plant Board. Next, identification of these priority areas, especially in the research in Ayurveda. So here, actually, this is a flow chart. I may not be as clear in this. Anyway, I will explain to you more clearly. So first and foremost, identification of this priority area. So there are, as per the IMR and EMR, different priority areas are there. So extramural and intramural are different priority areas. They will, if you go to the site, they will give the list of priority areas. What are the diseases they have to take, and apart from the disease, for the sake of public health, there are different priorities. I will explain these priority areas also. So first, identifying this priority area. Next, that is followed by with the literature of survey of the literary review or the literature and the previous knowledge that is available, that we need to survey on this, and the hypothetical basis, like either it is a single drug component or compound formulation, that is hypothetical. I mean, hypothetical basis. On this basis, we need to hypothesis of this, and drug for identification of the identified condition. Drugs for identified conditions like a standard operating procedures, preclinical safety or biological activities. So the conditions we need to identify them, and the specific protocols for the research methodology, specific protocols, and this being identified and approved by the task force from the funding agencies. Those who are giving the funding, they will send some task force, either the required research institutions, I mean, proposed research institutions are having required infrastructure or not, then only they will allow this funding process. So the approval by the task force, and finally they will come to the clinical trial, right? So this is the flow chart of this, how the research is being conducted in the IMR or PMR. Now we are talking about the apex body for this research. So we have only this. So the government of India set up this CCRS as an apex body, and what they do the activities of formulation, coordination, development, and promotion of the research in Ayurveda. Formulating the research and coordinating with the not only this Ayurveda, with collaboration with the other sciences and the research with the other medicines. System of medicine. Next, so here is the mission to develop quality assured and cost effective research products for health promotion, prevention, management, and to cure the disease, to meet the needs through establishing the facilities for comprehensive research with a focus on the public health sector. And this integration with the mainstreaming, like CCRS with ICMR collaboration. This is the present combination of these two organizations having some basic objectives, a mainstreaming of the practices of ISM, that is Indian System of Medicine, like Siddha, Yoga, Naturopathy, and Ayurveda. So promoting this child health and the reproductive and child health programs, primary health centers. In the PHC level, they are mainstreaming. So each and every PHC will be co-located with this Ayush department. and Ayush dispensary will be established in this every PHC level, and focus on the prevention and management of the common conditions that lead to morbidity and mortality during antenatal and postnatal period. So, and acute and chronic toxicity studies, toxicological studies. So these are all the main objectives, some of the main objectives, and there are other objectives also. So I have just shortlisted those objectives. And the organizations which are conducting this research in this field. So that is all the PG institutions, that is PG, they are conducting the research as per their institutional researches, and CCRS, ICMR, CDRI, AIMS, NIMHANS, and DST, CSRI, IGBA. I'll explain to this. So ICMR, we all know that is the Indian Council of Medicine Research, and CDS, Central Drug Research Institute, AIMS, All India Institute of Medical Sciences, that we are very well known about this. NIMHANS, that is the National Institute of Mental Health and Neurosciences. It is also collaborated with Neuro Health, Neuroscience, National Institute of Mental Health and Neurosciences. So DST, that is the Department of Science and Technology. Next, CSIR, that is the Council of Scientific Industrial Research, Scientific and Industrial Research. IGIB, a small correction, Institute of Genomics and Integrative Biology. These are the collaborative organizations which are combinedly working together in the field of research in Ayurveda. And these are the priority areas of research. So these are the research as per the IMR, that is a individual effects of each individual therapy, efficacy, safety, and of the individual drug and individual diseases, and research into mechanism of action of individual therapies. So the procedures, treatment therapies, and including patterns of the response to the treatment. And research into strategies of, and the research of finding the new research strategies in the traditional medicine, and research into efficacy of the diagnostic methods, and research focus on the refractory and the chronic disease. So these are about the AMR priority areas and EMR priority areas. I have enlisted this. I will share this PPT so that you can go through this. I want to read all this. So there are several diseases, and especially these are the lifestyle disorders. And these are the lifestyle disorders and the chronic disorders where they are giving utmost priority for the research. And the core areas of the research, that is the medicinal plant research, drug development, and literary research and documentation. And there are some broad areas here. So the development of methods and modalities for Ayurvedic clinical research and development of protocol for clinical trials, epidemiological research, preventive health, and research and development on Ayurvedic diagnosis. So including Nadi Pariksha. Actually, even it will take a diploma or degree course. Just if you take, this is one of the concept in Ayurveda, Nadi Pariksha, examination of pulse. A simple pulse examination will be taken as a diploma course. Okay, even it is also being conducted in so many North India institutions, central institutions, and autonomous institutions. So one single concept is enough for a diploma, means you can understand as many as diagnostic tools are there, and they are also every concept and every tool should be given in a new research mode. And research and development on, especially on Panchakarma, as it is a hot trendy therapies everywhere they follow, and the applications of research in Ayurveda. And here are some of the applications like first line therapy, adjuvant therapy, rational polytherapy, protective therapy, and economic therapy. So economic therapy is also one of the line of this applied aspect. So to reduce the dose and cost of this treatment aspect as well as the drug. And to the protective aspect is to prevent and treat the adverse effects, and for the as a primary therapy. and the rational polytherapy for individualized management, and adjuvant therapy to improve the response of the primary therapy. So these are the applications. And next, drug research and development. What is happening in the, so these all comes under the different research activities. So modern herbal research, that is about the National Institute of Tropical Medicine and SEMR and CDRA, they are screening the medicinal plants for their biological activity. More than 2,000 plants already been screened. Sorry. And drug research and development. So here is standardization of the drug, pharmaceutical research and development, pharmacognosy, both in vivo and in vitro. Biomolecular and biomarker based mechanism of the reaction. So we are taking DNA testing and DNA sampling for these medicinal plants, and ethnobotanical research, survey and documentation of medicinal plants and veterinary Ayurvedic products. So these are all the drug is R&D. R&D as in a glance in Ayurveda, medico ethnobotanical survey. So I will show this image here. This is medico ethnobotanical survey. So that means they are digitalizing this herbarium of all the medicinal plants, those who are going to extinct and endanger, and those who need to promote in a broad scale. So next, tribal healthcare research. This is approximately 2004 claims folklore medicine was claimed here, and pharmacognostical standards, pharmacological studies, the pharmacognostical 207 in the API, Ayurvedic Pharmacopoeia of India, they have given standardization for this pharmacognostical studies of this medicinal plant 207, and pharmacological study, that is about 375 drugs approximately, and physicochemical standards. These are like 631 drugs and 841 formulations are given in the AF5, that is Ayurvedic Formulary of India, and several publications. So this is the Ayurvedic research database that is only about the books, not about the articles. These are the books that is published here. I will show one by one with the pictures, images. So this is the medico ethnobotanical survey. So the survey tours, 1223 tours and areas surveyed. This is the statistics showing about this the survey. Next, yeah, coming to the tools, diagnostic tools and the therapeutic tools. So this is one of the example, automated Sarvangadhara Yantra. So Sarvanga Dhara means it is a continuous flow of the medicated ghee or oil in a particular area of the body. So that will be giving some real absorption and deep absorption of that medicine as long as it is poured in a continuous and with the right amount of time and the right amount of dose. So this is what they have created is automated yantra. So the person will be stable at one point, the whole body. This is Sarvangadhara. That means Sarvanga means the whole body. So one cannot hold this if you are using for 1 hour. So continuous pouring of this medicine for an hour. Nobody can hold this for 1 hour. So what does do? It is a automatic moving of this Sarvangadhara to cover the entire body. So this is one of the biomedical and instrumentation research that is being done. And not only this, preparation of the Kshara Sutra in most sanitized areas and the sanitized and easy, quick preparation of this Kshara Sutra. Actually, the one Kshara Sutra will take 27 quotes, one by one, 27 quotes, and even the preparation of we have a fine. I will give you an example. Ekottara will be there. Taila. So one taila, that means Setottara, Sahasrottara, that will give, that means 100 times. So I will give one simple ratio. To prepare an oil, we will take the ratio of 1:4:16. That is 1 part of dravya and 4 part of oil and 16 parts of kwada. Okay. So So if you are taking 1 liter of oil, we require for preparing a medicated oil. So that means 1/4 of the medicine paste or powder that has to be taken. And the oil base will be 1 liter, and that will be 16 times of decoction of that medicine. Medicinal decoction taken with 16 ratio, 16 portions. That means 16 liters will be there. And that will be evaporated to the reduced up to 1 liter. What we require is only 1 liter of the oil, right? Medicated oil. So for that 1 liter, you are adding 16 times of decoction. All the decoction should be, absorbed into this oil, and the medicine is also absorbed. So that is one time preparation of the Taila Paka Vidhi. It's the right procedure of preparing a Taila. Now I'm talking about Satottara Sahasrotra. Such a way you have to do 100 times and 1,000 times. That means you need to evaporate the decoction. Every time you will add the decoction and you will reduce to the Taila. So such an intensely, highly qualified preparation. So these kind of, it will take how much time to prepare such a kind of Taila. So all these being automated, and we are introducing some new techniques for preparing different formulations. Okay, so that is about the instrumentation and the therapeutics, and as well as the medicinal formula preparations and formulations. Now talking about the literary research and documentation. So the books, the CCRS has published 239 books, and there are e-books. So these are the, when it was started in 2006, 8, I'm not very sure, around before 2010, it was started giving this research portal, and that is about the e-books, electronic books. So that is on. So these are about the classical which have been translated in multilingual, like Cheraka Samhita, Susudaka Samhita, all these Nigantus and Samhitas been translated, and they are open access for one and all, with the multiple languages. Right? So at first they have started with the 7 e-books. There are number of books are being started now, and microfilms available in this council, that is around 272, and within the headquarters there are 135. And this Heritage, National Institute of Indian Medicinal Heritage, that is in Hyderabad, and they have some of the rest of the other books, that is 137. So these are all the literary research and documentation that is ongoing in the research activities, and the availability of this electronic and published database. So here we have a CCRS database and CSIR format in the book format as well as the electronic format, ICMR database, and NAM-STP here also comes under this, and Rudra, this is a software-based data on the basis of Ayurveda. So here is the researches in Ayurveda, a classified directory of PG and PhD thesis. And here PG and PhD thesis are not accessible, the entire thesis, only the titles and the full thesis are not available, but Shodhaganga. So this is UGC sponsored based for PhD thesis, which are continuing titles, even the full thesis also. But if you want to go through this entire PG thesis of the autonomous universities, and every state has got university except this Andhra Pradesh, but all the state government universities, they will have this research database and Ayurveda research database. It is also available in the CDs and the online electronic. So you need to subscribe this so that you can easily assess this thesis and research books. And electronic book publications. These are the list of these publications. And intramural. So what are the intramural clinical trials that are being taken in this aspects? That is about osteoarthritis, rheumatoid arthritis, osteoporosis, obesity, diabetes mellitus, bronchial asthma. So these are the lifestyle disorders and the chronic disorders where they are taking this intramural clinical trials. And this is the extramural research. So developing the research and development based Ayurveda drugs and to generate data on safety, standardization, quality control, And here are this research, extramural research priority areas. CCRS schemes, already I told you about this. There are several funding, and the most important and most interested aspect, that is funding agencies. So the Department of Ayush, the Ayush Ministry, is having funding system of those who are conducting the research in the state councils and the central council. Okay, next, central council research, already given IMR and EMR. So even if you want to do some extramural research with the collaboration of this Ayurvedic colleges and institutions, the least minimum will be starts with 10 lakhs to up to 3 crores. That is the minor projects. In the major products, will have the highest finding, and also as per the flowchart of the research, they will follow all this the investigators, the principal investigation, co-investigators, and the verification of the infrastructure, and even they have some guidelines and norms even for the area and plenty area of the clinical observation or examination of the patient room and the patient bed. So each and everything as per the norms, and if the infrastructure is as per the norms, it is easy to get the research project and acceptance and approval of the research projects. And the National Medicinal Plan Board, already we discussed about this, and the Department Biotechnology, Department Science and Technology, and ICMR, Indian Council of Medical Research, which has given funding for this preparation of I-64 recently. So these are the different funding agencies. So now we come to the great Golden Triangle Partnership. Actually, the image was, there is a triangle in between this, so it is not, I don't think it is clearly visible to you. So that is Ayush Research Councils, whatever I have mentioned, the Central Council and the statewide councils, and ICMR and CSIR. This is a great combination of, this is what we call GTP, Golden Triangle Partnership. So the objectives of this GTP, so they are looking forward to give this national-wide large scale projects and research projects. So the objectives of the GTP is to bring safe, effective, and standardized ASU products. Okay, and to develop new products effective in the disease condition, and to utilize appropriate technologies for development of single and polyhedral products, and to promote the collaborative research on the Ayush Ministry and the Ayush Department. So these are the objectives of the GTP. And here I want to conclude with this, the noble contribution of Ayurveda in the modern day health scenario. So there are almost all the WHO given this in this era, the lifestyle disorders, the Panchakarma and Rasayana therapy, the immune therapy of Ayurveda is giving a hope for healthcare, and it is being accepted by the WHO also. Right, and it is giving the preventive aspect of the diseases like the metabolic disorders, genetic disorders, and autoimmune disorders, and seasonal diseases, and aging and geriatric. This is about the preventive aspect. And alternative therapies like in the allergic disorders, like collagen disorders and Parkinson's disease, Alzheimer's disease, and so many several skin diseases and gastrointestinal disorders. This is as an alternative therapy. And a complementary area of cooperation, that is in the cancer, diabetes, ischemic heart disease, and cardiovascular arrest. So these are having some complementary area of cooperation in this bronchial asthma, neuromuscular disorders, and psychosomatic diseases, stress management, and stress-related syndromes, ulcerative colitis, IBS, psychological disorders, anxiety neurosis, sleeping disorders, and musculoskeletal disorders, joint disorders. So these are giving this some promising results in these 4 aspects of this. This is a great contribution of Ayurveda as per the present health scenario. And so sum up all these things, world is eagerly waiting for the help of Ayurveda in solving the health problems, and the government of India is already ready, and it is offering so many financial help for the development of Ayurveda, not only nationally, even in an international brand also. And all the stakeholders of Ayurveda, the researchers, practitioners, teachers, lawmakers, professional organizations, all the practitioners have to respond to this to call to for the health scenario. And the future Ayurveda, it offers a lot for personal and professional development. And it is the only way, the only practical way to inbuild scientifically any medical system to acquire global acceptance and for health for all. Health for all, that is the only practical way is possible scientifically without any and global global acceptance. So here are the research what is adding to health system. So with this I conclude here. So do you have any queries regarding this until now? If you have anything, you please feel free to ask the questions. I know it is a bit late, but still
Good
if you have
afternoon.
got any doubts. Yes,
Good afternoon, ma'am.
yes, yes, madam. May I
Good
know?
afternoon. My
Can
name
you
is
please introduce?
my name is Dr. P. Aparna. I'm a faculty member here in Vijaya Pharmaceuticals, ma'am.
Namaste, madam.
Namaste, madam. It was really nice, ma'am, because regarding the research of Ayurveda, you have given a really, I have been following it from the beginning. It was really a nice presentation, ma'am. We came to know about so many things because nowadays this medicines for COVID and all are coming into
Yeah.
picture, madam. So
Yeah. Actually,
was
I
a bit,
haven't covered the area of COVID because it is a very large,
yeah, I know.
much
I know.
time. So I haven't touched that point. That point, if we discuss, it will take hours together.
Yeah.
So I haven't touched that point. Right.
Yeah, because
Yeah,
we
but-
people are getting into touch with these things, and
Yeah.
I have one doubt, ma'am, regarding not exactly
please.
the research line, but
Yeah,
one
please
doubt
go
I
ahead.
have regarding this.
Yeah.
Somewhere I heard that do you use any heavy metals in the processing of these Ayurvedic medicines that will be marketed in the market?
Absolutely. Actually, I'm not talking about the patent because the patent preparations are all coming from the pharmaceutical companies, which are with the collaboration of this modern medicine and all these things. We are not sure about it, but whatever the classical formulations, which are dealt in the classics, and what we are following the preparation of these medicines, definitely we will use the heavy metals like arsenic is there and mercury is there. So, but remember, so the main important point is these are being used as in a purified form. There are, I will give you some of the examples. Just to take Parada. So we have this Parada purification, Shodhana, Samanya Shodhana, and Visishta Shodhana. That means specialized purification and a general line of purification. Okay. In the general line of purification, we follow 8 methods. Okay, in the specialized method, we'll follow 18 method. Ashtadasa samskara. Samskara means which the drug being modified for better utility.
Okay.
And with a better efficacy. That is being nullified with all its toxicity. Removal of the, so we will use the Visha, absolutely you're right. It is a toxin, and we are using this Visha in a purified form as it is acting as Amrita.
Okay.
So
will it,
we are
extension of that doubt. Will it not be any accumulation in the body or anything like that? Heavy metal accumulation?
No. It is not. It will not be like that.
Okay.
So this will be taken in the form of basmas, so that will be not absorbed and not giving any adversity in the cellular level. Okay. And when we are talking about the first pass metabolism, so at this level it is not causing any effect with this portal circulation or renal water circulation. So, and one more thing, so these basmas we have this testing for these basmas also. Okay. So then only we will use this in a right method. And very important point is the fine doses. Very fine. micro and micro level doses, minute and micro level doses. So it will not be causing, but you should be remember about that. Even Amruta, so what I conclude, even if you are taking a divine nectar also, in excess dose, it will cause visha.
Yeah.
So the dosage will be very important. So that means, though it is a medicine, that should not be given in excess, right?
Yes.
Even a medicine is will be having
overdose is there even in it's
anywhere it is there.
medication also.
So
Yes.
so we will take only the micrograms when compared, and that is not alone with one that basma. It will be taken with us the combination of the mineral and mineral herbal and herbal. We have three combinations.
Not
Like
be a
herbal,
toxic material.
yeah, purely not and purely herbal, mineral, herbo mineral. So that will be nullified even if it is having less toxicity also.
Okay.
Even
Thank
after
you. Thank you.
yeah,
Thank you.
thank you so much. Still, if you have any doubts, please. Anyone else? Okay, thank you.
Participants, if you have any queries, you can. Students,
Yeah, I would like to get the queries from the students, as you might have so many doubts as on today. If not, I'll continue. So I would like to acknowledge my thankfulness to Principal Madam Professor K. Padmalata Garu and the program coordinator Dr. S. Praveen Garu, and extend my special thanks to Jairam Garu. He is my well-wisher and friend, and my professor and HOD Dr. P. Suvanajyoti Garu, who she given me this option. And I would like to thank each and everyone who have given this opportunity to share my knowledge and take views from you. And I would like to get the feedback from you. Thank you so much. Thank you one and all. Have a great day. We can communicate on after this session also afterwards, and you can come to our college. You can make a visit, and you have any doubts, you can clarify with the other specialists which are available in this department or college. Okay, thank you.
thank you, ma'am.
Thank you. Yeah, thank
That
you so
was
much.
really a very knowledgeable session and elaborate presentation. And
Thank
just like anything in excess is poison, but it's not like that. The knowledge and the expertise you have shared with us today is going to act like nectar if utilized properly in future by our student delegates. Hope they will make use of your session and come to you for further research, madam. Thank you very much for obliging our request and accepting our invitation.
Thank you so much, ma'am. Thank you. Thank you so
Thank
much.
you. Students will join for tomorrow's session sharp at 10 o'clock. I request you all, now you can leave the session and join us tomorrow morning. Thank you.